गणदैवताय गणाध्यक्षाय धीमहि गुणशरीराय गुणमण्डिताय गुणेशानाय धीमहि गुणातीताय गुणाधीशाय गुणप्रविष्टाय धीमहि एकदन्ताय वक्रतुण्डाय गौरीतनयाय धीमहि गजेशानाय भालचन्द्राय श्रीगणेशाय धीमहि एकदन्ताय वक्रतुण्डाय गौरीतनयाय धीमहि गजेशानाय भालचन्द्राय श्रीगणेशाय धीमहि।
A very good morning, one and all. I, Rupa Singh, Assistant Professor in Department of Pharmacology in KSP, take this honor to extend a very warm and hearty welcome to everyone present here. I request all the dignitaries to take their seat on the dais. To follow our holy tradition, to worship our goddess Saraswati before any event, I invite all the dignitaries to do the honor of lighting the lamp with a prayer.
या कुन्देदु तुषारहारधवला या शुभवस्त्राशामृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्कर प्रभृतिभिर्देवैः सदा वन्दिता क्षामान् पातु सरस्वती भगवती निःशेषजाड्यापहा निःशेषजाड्यापहा.
I once again welcome our dignitaries. I request our Registrar Sir, Dr. Chirag Nagra Sir, to welcome our guest of honor, Professor B.N. Suhagia Sir, with the flowers. Thank you, sir. I request our Director Sir of KSP, Dr. Falgun Mehta Sir, to welcome Professor Mehul Patel Sir with the flowers. Thank you, sir. Today, we have gathered here for this brainstorming and enlightening scientific session of Indo-US International Conference on Global Trends and Global Trends and Challenges in Drug Design, Nanotechnology and Pharmaceutical Sciences. I would like to invite our distinguished Director and Professor of KSP, Dr. Falgun Mehta Sir, to commence the event by extending a warm welcome to our guests and participants from all across the India.
Good morning, everybody. Are you all excited about the event? Great. So, I have been given this task of welcoming all the guests, and I would like to begin by paying my respects to the guests on the dais and the guests of the dais. The delegates are also guests for us, so we welcome all the delegates who are here offline and those delegates who are online with us. For those who are not aware, this session, this conference is entirely being held in a hybrid mode, which means that there are a few students, few delegates who are here offline, but we have a lot of other delegates who have joined online via YouTube. So there is a live streaming that is happening right now, and a lot of delegates are joining us online. I have a few statistics, numbers rather, which I'd like to go through for the benefit of all those who are present here. So we have participation from a lot of states. I will just name those states. Gujarat, of course. Then we have Haryana, Himachal Pradesh, Karnataka, Madhya Pradesh, Maharashtra, Punjab, Rajasthan, Tamil Nadu, Telangana, Uttar Pradesh, Uttarakhand, West Bengal. Dadra and Nagar Haveli, and we have 2 registrations from people who are currently in Australia. So this is the reach that we have had because of technology. A couple of years ago, maybe this would have sounded impossible, but here we are in this age and time where technology makes it also easy for us to manage. An event like this has a couple of agenda, a couple of points that we think of when we arrange such a conference. One of them being that the students who are currently pursuing their B.Pharm or their M.Pharm get an opportunity to showcase whatever work they are doing. Also, they get to know what others are doing. And this way they can improve their performance, they can improve their research, get new ideas, interact with people, network with people. Those who are in the profession right now will know the importance of networking, and hopefully you as delegates will also realize the importance of networking and knowing people. You can approach them for help, you can approach them for ideas, you can approach them for a lot many things, right? So this is one of the aims of organizing such a conference. Another is a lot of PhD research scholars have this, let's say, demand, a requirement of fulfilling poster presentations or publications before they are awarded their degree. And such conferences again will give you an opportunity to showcase your work. Maybe you can show it to your guide, maybe you can show it to the university from where you are pursuing your PhD, and that will also help you. Another major point, agenda that is covered in such a conference is interacting with and knowing or discussions with people who are stalwarts in their field or work area, be it from other states of India or from abroad. Like today, we will find out soon, we have one speaker who is from abroad and we have 2 other speakers who are from Gujarat and another state. So that is another agenda that is fulfilled by such a conference. I would like to, I know this is an inaugural session, but I would still like to thank the faculty members, the student volunteers and the delegates and the speakers for being here and giving us the honor of receiving them as guests. And we look forward to a lot more interactions with them in the future and with the delegates in the future also. Thank you so much.
Thank you so much, sir, for your insightful words. I would like to extend an invitation to our special guest of honor, Professor Suhagya sir, who is a Chief Advisor in APP Gujarat State Branch and Research Director in Dharam Singh Desai University, Nadyad, Gujarat, to grace today's event with his wisdom and thoughtful words.
Good morning to one and all. Respected Sri Jagdish Bhai Patel, President of KPGU, Sri A.P. Chaudhary Sahab, Provost of KPGU, Sri Chirag Nagda, Registrar and Control of Examination, the management and staff member, and also thanks for arranging this type of conference, Indo-US International Conference on global trends and challenges in drug design, nanotechnology and pharmaceutical science. It also gives me great pleasure to be here in presence of this event in front of young budding students and scientists. So it is a very good session and I got this opportunity to say a few words. The topic is rightly chosen, drug discovery, nanotechnology and pharmaceutical science. Drug discoveries refers to the process of designing novel drugs, and that takes approximately 10 to 12 years. Now, after nanotechnology is a bridge for biological and physical science by application of nanotechnology at various fields. So, we have preferred nanotechnology rather than the discovery of new drug entity. So we have changed nowadays the drug delivery system also. There are certain challenges that needs to be addressed to facilitate advancement in drug targeting. A lot of research in the field of last 2 decades has already led to filing approximately 1500 patents on nanotechnology and completion of several dozens of clinical trials. So, thanks to advance in nanotechnology and nanomedicine, our ability to diagnose disease and even combining diagnosis with therapy has also become reality. Now, era goes toward the application of artificial intelligence that may become part of everyday modern life, and new biotechs are advancing into AI facilitated drug design, and in real world outcome and become potential in life science industry. AI is successfully used to identify potential drug used in COVID-19. Google DeepMind AI system used in AlphaFold has found solution to how protein fold in 3D structure. So this may create the opportunities in drug design. So by finding the protein structure of various bacteria, virus, we are able to prepare a drug for that particular disease. Now, after that, I will talk about some few words about the APP. So APP is a great platform for young pharmacy professional to share and exchange ideas and boosting the young minds. With this, I wish all of you a great experience in this one-day seminar, and I hope that it would knowledge enriching and would create this spark in you to learn and deliver the science of drug discovery. I also congratulate Professor Alekha Das for untiring support to APP conference worldwide, and I also congratulate Saurabh Kumar Banerjee coming from the Rajasthan, and I extend my sincere thanks to Dr. Rajiv Dahiya and his team, and I salute to the dedication towards APP. So all the APP branch conferences in abroad and India are managed by G. Rajiv Dahiya, and he is taking too much strain for this type of conferences. So we appreciate them, and I congratulate and appreciate all awardees to get APP awards at this international conference. And also, before conclude my session, I want to do one announcement. Dr. Falgun Mehta, Principal and Director of Krishna School for Pharmacy, now from today he is nominated as a Joint Secretary for APP. Please give a big hand. And last, I thanks organizer for inviting me in this event and share my views. Thank you.
Thank you so much, sir, for your thoughtful words. Now I request Dr. Falgun, sir, to give the memento to Suhagya, sir. Event with the Students' Award Ceremony. It is time to honor and celebrate the exceptional talent that we will witness today. I would like to invite Professor Suhagya, sir, to give away the awards. The award for the best B.Pharm student in male goes to third sem student Sumukh Vasisht. Next award is for Best B.Pharm student in male goes to Janvi Surti. Next award is from B.Pharm 6th sem student is Rishi Kumar. Our next award is from B.Pharm 6th sem for Best B.Pharm student in female goes to Ashka Desai. Thank you so much, sir. Now I would like to invite Mehul sir to give away the other awards. Next award is from B.Pharm 8 student for Best Student in Female goes to Vrushti Shah. Our next award is from B.Pharm 8 SEM for student Best Student in Male category goes to Vivek Pradhan. Our next award is from M.Pharm SEM 1 for Best Male in for Krupal Shah, Krupal Sadhu. Our next award is from M.Pharm SEM 4 student in pharmaceutics for a female, that is Anuragvanti Thakkar. Next award is for the best M.Pharm student in pharmaceutical technology in male goes to Yuvraj Patel. Thank you, sir. Now it's time to give awards to teacher. I would like to invite Registrar sir to present the award further. The best young teacher award goes to Dhairyashri Kher. Next award, the best young pharmacist award goes to Miss Sanjokta Soni. Best young talent award goes to Meghra Suryavanshi. Our next award is for the best young teacher goes to Tawfiq Mullah. Thank you, sir. I would like to invite Falgun sir to give away the other awards. Young Achiever Award goes to Imtiaz Bagban. Our next award is for Best Talent Award goes to Parul Wasawa. Our next award is for Best Research Scholar Award goes to Mariam Mandarawala. Now it's time to give Best Teacher Award, which goes to Ashwini Patel. Thank you, sir. I would like to invite our Suhagya sir again. The Best Young Performance Award goes to Dr. Upasna Yadav. उधर से बोलो ना भाविनी को. The next award is for the Best Researcher Award goes to Kuldeep Pinchurkar. The next award is for the Best Pharmacist Award goes to Jigar Vyas sir. Our next award is for the Best Achiever Award goes to Dr. Sailesh Korodia sir. Our last award, this award is for the Appreciation Award which goes to our one and only Director sir, Dr. Falgun Mehta sir. Thank you sir. Sir, continue कर रही हूँ. I would like to request Dr. Chirag sir to give token of appreciation. Sorry, Dr. Jigar sir to give token of appreciation to Chirag Nagra sir. Sir. I would like to request Mihir sir to give token of appreciation to Suhagya sir. Thank you.
Thank you, Rupa Madam. Respected dignitaries sitting on the dais, directors of various schools, HODs, faculty members, dear students and delegates. A very good morning to all. Myself, Dr. Sailesh Korodia, Professor at Krishna School of Pharmacy and Research, Drs. Kiran and Pallavi Patel, Global University, Vadodara. Feel honors to get an opportunity to propose a vote of thanks on this 31st Indo-US International Conference. My sincere thanks to Professor Rajiv Dahiya, Scientific Committee Chairman and Founder President, APP, and Dr. Sunita Dahiya, Organizing Chairman and General Secretary, APP, for giving us an opportunity to organize 31st Indo-US International Conference. My special thanks to our guest of honor, Dr. B.N. Suhagya sir, Research Director, Faculty of Pharmacy, Dharamsi Desai University, Nadiyad, Gujarat, for accepting our invitation and for being here with us today. Sir, your thoughts have truly inspired us. My heartfelt thanks to our Honorable President Sir, Sri Jagdish Bhai D. Patel, our Honorable Vice President, Srimati Kamalben J. Patel, our Chairman Sir, Sri Dhruv Kumar J. Patel, and our Secretary Sir, Sri Krishna J. Patel, for the motivation and valuable guidance. My sincere thanks to all the speakers of scientific session for accepting our invitation and sharing their valuable knowledge with the delegates. My special thanks to all jury members for accepting our invitation and evaluation of posters in hybrid mode. I would like to thank our beloved Director Sir, Dr. Falgun Mehta, our Provost Sir, Dr. A.B. Chaudhary Sir, our Registrar Sir, Dr. Chirag Nagda Sir, our OSD Madam, Ms. Palak Landge, Deputy Director, Deputy Registrar Academics, Dr. R.K. Yadav Sir, HODs, faculty members, non-teaching staff for their constant support and to shape the conference. My special thanks and wide round of applause to all the delegates who made conference a grand success. Last but not least, a big thank you to each one of you who directly or indirectly made this conference memorable to all of us. Thank you very much.
Thank you so much, sir. Now I request all the dignitaries to sit off the dais. Vivek, chair. हाँ सर, वो chair वगैरह हाँ. The inaugural session is over. Let's commence the event with the technical session by our eminent speakers. I request Professor Alekha Das sir to take his seat on the dais. Now I request Dr. Jigar Vyas sir to welcome Alekha Das sir with a bouquet. Our first lecture is on the cancer nanomedicines advances and challenges. Nanomedicines are applied alternative treatment for the anti-cancer agents for the treatment of cancer due to small size in nanometers. Specific site targeting can be achieved with the use of nanomedicines. To provide expertise on this subject, we have a highly distinguished individual, Professor Alekha Das sir, Associate Dean, Department of Pharmacy Sciences, School of Pharmacy and Health Profession, Creighton University, USA. To introduce him, let me invite Dr. Jigar Vyas sir, Professor in KSP, KPGU.
Thank you, madam. Well, it's my privilege to introduce our first technical session and first speaker, Dr. Alekha Das. He has received his bachelor and masters from Jadavpur University in 1981 and 83 respectively. He has completed his doctorate from Minnesota University in 1990, and he has been a member of Creighton University School of Pharmacy and Health Professional dosage forms for nutraceuticals. He has been involved in teaching various subjects of pharmaceutics, pharmaceutical analysis, physical pharmacy, industrial pharmacy, and many independent research. Dr. Das has received several awards during teaching, research, and his service. He has, to his credit, more than 50 research papers in various renowned international journals. He has few patents. He is a member of many professional bodies. He serves as a member of editorial boards and reviewer panel in many renowned journals. He has, to his credit, chapters in various textbooks, and he has delivered such technical sessions before today. As a part of sponsored research, he has worked with many industries like Phenolic Corporation, Omaha, Surgeon Pet Care's product, Alliance Laboratories, Thermolife International. He has also received Academic Excellence Award in 2019 by Association of Pharmacy Professionals, that is APP, in APP's 7th Indo-US Conference, which was held in Jaipur. He has also offered various consulting service in Armenia, USAID during 2013, Department of Pharmaceutics, Faculty of Pharmacy, Kuwait University in 2012, Healthcare Pharmacy Technology in 2008 to 10. For his valuable contribution to the department where he was working, he has received Omaha Family Foundation Awards several times. So I heartily welcome Dr. Das sir to this technical session, and I request sir to please continue with today's sessions.
Well, thank you. Thank you for the invitation. Thank you, guests, delegates, and especially my students. And my attraction is not for anybody else rather than students, because that's one of the things I like most, and it's to my heart. I'm here whenever from APP. I also represent, I'm the president of American chapter of APP, and I have done many sessions here, and I have committed that anytime I'll be visiting India, I'll be spending some time for my profession. Even though I was introduced, I have my bachelor's and master's from this country, but very few people knew that I started, actually my lowest education was in diploma in pharmacy. So I'm a diploma in pharmacy graduate. And today, what I am, it is because, two things I want to give it to you, especially the students. The take-home message I want to give: nothing is impossible in this world. If you have the motivation, if you have the courage and determination, anything is possible. And that is an example to me. I decided this profession, I was talking to the director, that for a reason, I selected education because my mother didn't know how to read and write. So when I come from school, there is nobody at home to help me, to guide me if I have a question on any of the subject. And that was my start. And that is the reason I decided I could have earned lots of money in pharmaceutical industry with the work I do if I have taken pharmaceutical industry as my career, but I didn't. So that's the reason, the first things I was talking about, nothing is impossible. And the second thing I would like to see, some of you will be better than what we are today, especially in the field of research. In my definition of research, I don't know how many people know what the research is all about. As the name implies, it takes research, search again and again. I know that your institute, I was very impressed with your institute. I've seen many institutes in India, abroad, and many foreign countries. I've traveled tremendously in different places of the world. But one of the things I think that you are so fortunate. You are so fortunate. That's one of the things we always forget about, that we don't have. This is one of the things always we complain. We don't have. But we don't never think about what we have. So that is one of the things I will tell my students. Even though you don't have an instrument, powder oxidized fractional in your lab, still don't do research without powder. You can send the samples, we can collaborate with the people, you can find out with all this information technology today, you can find out where there is a powder oxidized fraction instrument in this Gujarat. This is a state which is resourceful. I came from a state when there was no pharmacy school. I have to go to Calcutta to have my bachelor's degree. The only things I had, I was a very good student. That is the only things I had. Nothing else, nothing more. So if you have the determination with that, I'll start my talk today. Since I know that this is a bachelor's, master's, as well as PhD students, some of my esteemed faculty are here. I'll try to give not too much information here. There's lots of information there, but I'll be giving you some idea about what we do this is a master's project. So this will give some idea what in a USA, in a pharmaceutical institute, what type of work these master students do to have their master's degree at the end. Mostly it is research related. It's not like course. There are some schools that they only produce, you take some classes and you will get your, okay. So what I did is, in this particular story what I'm going to talk to you guys is, we talk also in the morning that how to put new wine in old bottle. Okay, that's the basic ideas. You know that drug development and discovery is a very lengthy process. You know that some of your faculty are discussing those in your curriculum. You know, it takes near about 7 to 18 years sometimes. And one of the things I also want to tell, because there are lots of good dignitaries, lots of administrative position, potential people here. I have only one regret. What I have left this country 45 years ago, where the profession was versus the profession I do see today, is in the same place. A little bit ahead, but we have not, this profession of pharmacy has not got there. Again, we have not received what we can do for society, what we can do for humanity, what we can do. And one of the classic examples the world has seen that what a pharmaceutical scientist or a pharmacist like us can do and save millions of lives. Anybody heard about this COVID virus? And the COVID virus, the vaccine we talked about, this was contribution by one of your pharmaceutical scientists who introduced this fact, that means the solid lipid nanoparticles can incorporate this mRNA. And because of that platform, this particular vaccine was actually approved within 3 to 4 months, which was a classic example what a pharmaceutical pharmacist or pharmaceutical scientist can do. So world has recognized us. Well, I can have many examples. We have also some negative examples I can give, because sometimes we always talk about positive things. Also, we have some to recognize the negative things. Think about diabetes. Okay, insulin was discovered in University of Toronto, 1942 or 40s. I don't know exactly that. I may be wrong, but it is around that time. But today, still, the patients are injecting themselves. We don't have a drug delivery system or dosage form. Even though we developed one, we took it out from the market because of toxicity reason. Hopefully, it will be coming again. But again, the patient has to take by injections. We have not have a delivery system to deliver this protein, which is, you know, 51 amino acids, because of the challenges. So the mother nature will be giving all the challenges. So we think that we achieve something, some homework will come from us. And this is another homework for us. In 1986, when nanotechnology, one of the, you might have heard about the EPR effect, which was discovered by some of the Japanese scientists in 1986. They think about the tumor versus normal cells. The tumor cells has leaky vasculature, normal cells doesn't have leaky vasculature. So they thought we got the magic bullet. We can make the drug in a nano formulation, so it can go to the leaky vasculature, it can go to the tumor, and tumor doesn't have any lymphatic systems, so it cannot be eliminated. So it will stay there, kill the tumor cells, but normal cell will not be affected. So we are very excited at the time, in 1986 I'm talking about, okay? I was a graduate student at that time, University of Minnesota. So when that thing happened, the mother nature has different stories. EPR effect, what we call extra permeability and retention, is not true for all tumors. It may be for some of the breast cancer, maybe some of the colon cancer, not for pancreatic cancer, not for other cancer. So basically, even though we think that we got something, we still have issues and problems. And this is one of the case studies I'll be providing you. This is a drug which has been approved for something else, but we are utilizing this drug, we call the repurposing. Repurposing is becoming a big word in FDA right now, because some of the drugs, and that is credit goes to all the scientists, the researchers, which talks about drug delivery research. Some of the drugs was not approved because of the problems, right? One of the problems was poor solubility. Aqueous bioavailability is less. It cannot be given orally. So when all these things, toxicity and everything has been done, and it failed finally because in the phase 3 trial or in the human trials. So those drugs are already there with the company. But what we are doing is, we are taking those drugs which has been already tested for toxicity and other purposes. Can we develop them into different doses form? It may not be orally effective, but can it go by nasal, by aerosolization, or by topical formulation, or by transdermal delivery? So that is a new trend you will be finding. That's the reason why I'm bringing this to some of our research students who are doing research, how to utilize some of the drugs which has been approved for other purposes, utilizing different formulation technology, how you can improve their bioavailability. And this is one of the examples. I hope I can advance my slides. So I put lots of background, so that's why I will be doing very quickly the stories, okay? So I'll not be sharing too much because I gave that I'll be finishing by 30 to 40 minutes. That's the time. So please help me 5 minutes before, just somebody has to let me know my timer. So because as a teacher, when we teach, I forget everything. I forget about my name also sometimes. So this is a topic, when you talk about nanomedicine, I don't want to go for nano, and you know what is nano is, very small, okay, micro, macroparticles, nanoparticles, macroparticles. So nano is a very smallest, you know that, I don't want to give the math to scare you off, how small it is, okay. These are small particles, right? When it is in the size range of near about maximum up to 500 nanometers or so, ideally 100 nanometer, you will be finding out it has some different properties. And we talk about one of the properties is EPR effect. That means because of the vasculature, they can easily pass through. But that is a passive way of targeting a tumor, passive way, right? But we can also manipulate, because we are pharmaceutical scientists. We know how to make the cookies. The cookies can be different types, okay? We can make the cookies depending on what the patient needs. So that's what we do. So you can do active the targeting. That means you can surface modify the nanoparticles so that if there is a particular target organ in the tumor cells that is overproducing a particular protein or peptide or whatever material, and you want to target it, you can do it. You can do by immunotechnology and so forth. So basically what you can see, whether it's a human system or not, when a nanoparticle is given by EPR effect, it's taken, this is the passive way of targeting. But if suppose by passing way you cannot target, remember I told you that some of the tumors doesn't have EPR effect. In those cases, this passive targeting is not going to work. Okay? So in that particular case, I'm not going to stop. I'll not lose my things. I'll be starting motivate and find out a way how to do. I have to take advantage of the environment of the tumor. It's different, right? Acidic environment and lots of other things. Salicylic acid will be produced a lot on the surface. So those will be negatively charged. So I can make positively charged particles. I can modify the surface of the particles and so forth with peptides and all these markers, right? So those will be considered as active targeting. So what I'm showing here is mostly the passive targeting, because this is just like a simple case studies, okay? So you know what is a breast cancer is. I don't want to talk about the things. It is just a tumor. It starts from a normal cells, but it mutated or something happened. So if it is not properly treated, as you know, that is the most important thing. Especially I'm worried about my fellow mates, because here people, if you have a problem, you don't go to a doctor quickly. You have to go to other avenues, homeopathy, allopathy, this and that, and quack, and so many things. And some people usually go to the last moment when there is a complication, right? That's where the problem starts, especially in case of cancer. Once the genesis will start, you see that it can migrate to different part of the bone and so and other process, and it causes all this, cardiac problems, brain, bones, livers, and so forth. Okay. So, and this is again, I don't want to go. This is just like a simple. It's not a 2024 statistics. It's a 2020 statistics. But you can see the importance of this disease. Okay. It is affecting many young people in the world, and this is one of the biggest challenge we have. And according to NIH, as you can see, that how many cases in US are near about 300,000 cases, which is an increase in 15% all new cancer cases in USA. So the treatment options available, as you can see, nowadays the survival is more. It's not because of the drugs or the dosage form of drug delivery system. I think most of the think it is going because of the early detection. That has become a very important thing by mammograms and other things, all the markers available, family history. You can track down the people who might be getting this disease ahead of time. And that is the reason why survival rate for this breast cancer treatment is increasing. Okay, so what are the treatment options? As I told you, you can do by surgery, chemotherapy, combination, and other things, right? This is one of the things we are talking about, chemotherapy. When you talk about chemotherapy, one of the major challenge of the mechanisms of the cell cycle, and this, you know that most of the cocktails or the drugs they are utilizing right now are less toxic as compared to 10 years ago, 15 years ago, and that's credit goes to pharmaceutical scientists. They found out the cocktails, what combinations, how to reduce down the toxicity of these compounds, so that it will not be toxic. Okay. One of the things I will be talking about, very quickly the biology. We didn't know about this thing 20 years ago. So, and that's why the treatment in breast cancer was really not very effective. When you are talking effective, now we know each and every individual is different. I was talking to one of the dignitaries there that sometimes we go for the West, but we never think about the East. But East has so much things to offer. We have so much vast culture. Our Sanatan Dharma, if you go back to the history, you know that I'm going to one of the Vedas where Ayurveda is a part of the Veda, and it talks about you know your patient. Categories of breast cancer. We didn't know that. So we are giving all these cancer agent to these patients. So if it is not the particular subtype, it is not going to work. Okay. So I just want to talk ER positive, HER positive, and TNBC. TNBC, which is called the triple negative breast cancer, and that is the most complex one. Unfortunately, Mother Nature has not given that much. It's only for a drug designer. That is one of the things we are working on right now. And this is one of the master students' work on this TNBC. Again, I'm not going to TNBC can be further classified into different categories. Again, risk factors. These are all, you can see it. I don't want to go into details. It depends on many factors like pathology, family history, and bone density, breast density, and so forth. There are different stages, medical field. That is one of the things I regret. That in this particular, especially in India, since pharmacist has not got the proper place, recognition that what a pharmaceutical scientist, a pharmacist, I'm a registered pharmacist from United States, and the respect that they give to a pharmacist, you cannot, it is beyond imagination and description. Okay, it is considered one of the most noble profession. Okay, and in the healthcare team, you are the only person who is expert in drugs. Outpatient will be calling them. Pharmacist has always to be called to find out what is the interaction, why it is happening. Do I have to change the medication right now? This antibiotic is not working. So they are considered as an expert in drugs in the healthcare team, and they are consulted. So if you talk about whether it is a physical assistant, physician, nurse staffs, dentist, or any healthcare profession, you are the only person in the team which is an expert on drugs. Look at this, at this age, I'm taking 5 to 6 medication at this age. All the population is trending to geriatric patients, right, of the world. So you as a pharmaceutical scientist or pharmacist has to deal with those patients, that you are in a profession that even though nobody is respecting you yet, but time will come, you will be well respected. But when you come, that respect, you know that there some people has got the awards. When awards comes, it comes with something else. You have to take that responsibility. So when there is a responsibility comes, as we have to, as a pharmaceutical professionals, please take responsibility to take our profession a little bit further away from this. Okay, I don't want to go for the stages of these things. You can see the TNM staging. T refers to the tumor size, and again, N refers to the proliferation of tumors, and M refers to the spread of the tumors per metastasis. So there are different scaling of these tumors. And why I'm bringing this issue here, I'm not a physician, but you as a pharmaceutical scientist or pharmacist has to know what are the stages, where the patient is, which drug will be the ideal drug for targeting. So additional markers of breast cancer are there. You know, there are grade 1, grade 2, and grade 3. And also there are some many subtypes. So these are the 4 medical, surgical management, radiotherapy, hormonal therapy, and chemotherapy. These are the 4 major treatment options available for our patients. Okay? So once you know which type of breast cancer it is, how will know? I'll know by diagnosis, by taking the blood samples, taking on the mammograms and all those type of things. We can able profile those patients from their genetic makeups to find out which type of breast cancer. Once we know it, then, so again, this is a list. This slide will be available for you guys. So you can have it and distribute. So I don't want to go for, so these are some of the commercial products available. But I'll be giving a little bit. Hopefully I have 10 minutes, right? Okay. So I'll be giving some examples. This is a drug which is called neratinib, which is used for the treatment. It's not approved yet for the treatment of triple negative breast cancer by nanocrystal formation. This drug is available as a crystal structure, just like crystalline compound. And you know, the crystalline compounds sometimes has problems, okay? So we wanted to change this crystalline structure of the compound to nanocrystals. Not formulation. I'm not doing anything to that thing. Rather, I'm taking the API, the drug, which is in this particular case is neratinib, to change it into a nanostructure. So triple negative breast cancer, again, this is a repetition of the same thing what I have told you before. Neratinib is a kinase inhibitor. You might have heard about. There are lots of kinase inhibitors for adult patients with early stage of HER2 type of, if you have a HER2 overexpressed breast cancer, that is the drug of choice, not for triple negative yet, right? So we are thinking that is this compound, if we'll be giving them as a nanocrystal, which will be taken by these things by your passive processes. NTB also irreversible binds with EFGR, okay, which is the critical things. We found that it can do that. If that is the case, so possibly we can utilize that property of this drug to advance them of triple negative breast cancer. So the objective was they started to the repurpose NTB for the treatment of TNBC by changing it from crystal to a nanocrystal. These are all the methods. I don't want to again go into all details of those type of things. I'll be available today completely. Anybody has any questions, I'll be interacting with you guys. I love to see your poster. I want to learn from you guys what you guys are doing. And if I have anything, any suggestions, I'll be providing. So one thing I have learned with over the years, and God has given us one mouth, but given us 2 ears. But this is for a reason. So we have to listen more rather than talking more, but we do the opposite. Okay, so that is one of the things. Whether you are a teacher, whether you are a student, try to listen, try to understand. You know, that will be giving you more learning opportunities. It's not obstacles. So these are some of the formulations we did. These are when you do a formulation or dosage form or drug delivery system or any device, do you know that what are the things we need as a formulation scientist? What are the 3 important things we want my drug delivery system or dosage form should have? I always do interactive. So any students, can you know that what are the 3 things we look for a dosage form or any drug delivery system or even stability? Well, it is a part of it. She started with S. S-E-R, not A-C-R, not S-A-R in medicinal chemistry people talk about. It should be safe, it should be effective, and reliability. It talks, that's why pharmaceutics is not pharmaceutics. It's only not formulation, analytical chemistry, medicinal chemistry. We have to make sure the drug contains exact amount of the drug and it works properly. The stability, one of our students talk about, Mudra, talk about the stability. That stability means you have to make sure the drug is reliable. That is what stability talks about, right? So these 3 important things we have to make sure. So for that reason, we have to do all these type of things called different types of big names, but simple things. You know, so a solution as well as you put in nanocrystals. Is it going more to the tumor cells when you put in nanocrystals? So if that is the case, it will be reducing the toxicity. Measure the toxicity. Toxicity can be many things, okay? You talk about physical toxicity. I'm not talking about physical toxicity. I'm talking about toxic cytotoxicity, because these are very, very toxic drugs we are exposing to normal cells and tumor cells, right, by injections. So we will be finding out, is it doing necrosis, or it is killing the cells by apoptotic ways. So those are the things we usually test, okay? I don't want, again, results will be available all with you guys. It has not been published. So please don't use this material for any other purposes, please. And so this is again, we found out by taking these nanocrystals. How do you make the nanocrystals then? Anybody know how to make from crystal to nanocrystal? Micronization, hammering. You have a big rock, and you want to make small strands. What do you have to do? Energy can neither be created nor be destroyed. It will be transferred from one to the other. So we apply mechanical energy to break the particles but what is the challenge? When you put this mechanical energy or electrical energy and breaking the particle, all the energy cannot be destroyed. It will be stored somewhere else. And it is stored as a surface-free energy. So these nanoparticles are very highly energetic active particles. And you know that the basic, you cannot be angry forever. You come back to the base level quickly. So all these energetic particles will not stay as energetic particles. They have to come to the ground state. So that is the challenge we have as a formulation scientist. How can you make them stable? Because these are high energetic particles. So that is one of the biggest challenge, and that challenge will be taken care here by utilizing some stabilizers. And we use some surfactants. You might have heard about what surfactant is. The dual character personality people around. They have both hydrophilic and hydrophobic personality, right? They can work with both sides. They're very good for, not for research, but they're very good for, in politics. They can work both dials, right? So you can see atomic force microscopy to see how the particles looks like. These are very nano sizes particles. We also did, this is a DSC, which stands for differential scanning colorimetry, where you can see the changes in the behavior of the particles when you heat the materials to see the physical changes that happens. And then you can have powder X-ray diffraction, because it's very important, because you are talking about crystals. If you give two powders to you as a pharmaceutical scientist, is it in crystalline or non-crystalline state? That is the only, I should not say only, but that is the most important technique available to us to make sure it is crystalline or non-crystalline, to find out. This is called powder X-ray diffraction. Most of the powder X-ray diffraction materials we'll talk about when you apply X-ray to crystalline materials, because of their long-range periodicity, you'll be finding a deflection of that light X-ray, right? And since they are packed very in a regular manner, you will get a diffraction, whereas in case of a non-crystalline or amorphous material, you will not get it. It will give a amorphous halo, okay? Again, XRD patterns. Then we have the aqueous equilibrium. Very interesting thing happens when the drug is present in crystalline form versus nano form. You see how much equilibrium solubility, 87-fold, 100-fold, very close to 100-fold increase in solubility, which will be enhancing the bioavailability and absorption, right? So again, physical stability we are talking about, the stability of the drug, because we are supposed to deliver this drug in injectable formulation. Okay, we will be presenting it as a solid, and people has to reconstitute and inject, right? So even though we have to show within 7 days, if the patient didn't take the first dose and within 7 days, is still stable or not. So the physical stability has been shown. Hemolytic potential is another thing, so we have to, I'll take 5 more minutes. Is it okay time-wise? Okay? Or if you want, I can stop it and we can discuss later. I don't want to override the timing. Is it okay for going for 5 minutes? So the hemolytic effect is when you put in IV systems, what is happening to the RBC? Is it completely hemolyzing this RBC or not? Okay, so that is one of the tests we usually do. Then we do the cytotoxicity. This MTT toxicity test will talk to you or tell you whether there is a necrosis of the drug. Okay, it will not giving you death will happening. Necrosis is not the same as apoptosis, right? So, to do that, we have to do flow cytometry. So these are some of the MTT toxicity studies results where we showed that if you give the drug in a nanocrystalline form, it is more cytotoxic as compared to your solution, which we wanted. Again, cellular uptake. You can do by confocal microscopy. You can see when you are giving the particles within a nano range, it is taken up by these cells within 2 minutes. That means cellular uptake is very quick, and toxicity is also very killing these the cancer cells quickly. Again, this is my final few slides about apoptotic cell death, to say that nanoparticles causes this cell death more as compared to solution. In summary, what I want to say that this particular drug, which has been approved for other purposes of cancer treatment, can be repurposed for utilizing in a nanocrystalline form, which has a better bioavailability, better safety and effectiveness as compared to the normal conventional formulation. With that, I want to acknowledge some of my fellow students and the postdocs, faculty friends who has helped us in other areas. As you know that, as I've told you, that you don't have to have all the instrument in your campus. You can still use it by collaborating with others. So if anybody needs any discussion on those part of the things, I'll be more than happy to discuss with you guys. With that, I'll stop it here. Sorry for my 5 minutes extra time. I'll be available if anybody has any questions. I'll be more than happy if I know the answer. Any questions, especially from my students? Yes, I'm not a genetic engineer, but yes, the answer is yes. I'm not a molecular, So one of the things I think I also learned that if you don't know something, nothing wrong in telling that I don't know the answer. Okay? But sometimes our students, I tell my students who comes for masters and PhD there, you don't have to know everything. Okay? Even your professor doesn't know everything. Okay? Even though they pretend that they know everything. Only the interaction between student and faculty. There versus here. So, well, things have changed now. So, no offense to anybody. I'm not offending any students or faculty here, but I do see that the interaction of student and teacher should be what our Sanatana Dharma talks about. You know that in Sanatana Dharma, again, this is where going back to the, since some Sanatana Dharmi delegate is here, I just want to impress him. But you know that Gurukula Ashram or Gurukul, when people are going to Dronacharya and all these people, they usually go with a purpose. Even though you came as a king or king's son, they will not accept you in that field. They work on strength, that called strength finder, what the United States uses to find out the different, what is your strength? If your strength is not for fighting, if I'll put in training for, that is useless my time. So my question here is, finding out the right people in the right bus is the responsibility of a guru, not a teacher. So gurus and teachers are different. So gurus are responsible for their day-to-day life till the end of their life. Even though they finished B Farm, M Farm, still then it is your responsibility to guide them so that they will be in their accolade. And that is your pride to be academician. So, thank you. I'll stop it here. Thank you again.
Thank you, sir. This was a wonderful session. And as per the title of our theme of the conference, sir has covered nano range. Means his talk was on nanoparticles, novel drug delivery system. And the session was very useful to one and all, especially me. I would like to associate myself with the work of our guest. As in during B.Pharm, sir, I work on the same topic, role of the gene therapy on the breast cancer. And during my masters, I have done in neosomes. There is nanotechnology. So for me, this session is very much associated. And sir has given nice talk, discussed since the beginning what is the reason, how the breast cancer, and the major part is in between he has also given many live lessons also, Sanatan Dharma, and the importance of Gurukula. So basically, I would like also to say that whatever you study in your life or in your career, if you are following some spiritual rules, you will be successful with whatever study you go in the market. So be there with your spirituals. Today, this is the over of the first session, and I would like to again invite Rupa Madam to continue with the next session. Thank you very much. Thank you, friends.
To continue the next session, I request Dr. Saurabh Kumar Banerjee, sir, to take the seat on the dais. Our second session is on career opportunities in pharmaceutical management for pharmacy graduate students. Pharmacy degree holders have a greater degree of flexibility than others because they either work as an entrepreneur or in an industry or in a hospital. We have an esteemed guest with us to talk about this subject, Dr. Saurabh Kumar Banerjee, Dean in School of Pharmaceutical Management at IIHMR University, Jaipur, Rajasthan. To introduce him, I would like to invite Dr. Prachi Pandey, ma'am, Professor, Deputy Director in Krishna Center of Startup and Entrepreneurship, KPGU.
A very good morning to all the dignitaries and delegates. A very warm welcome to Dr. Saurabh Banerjee, sir. And we are listening. We have just gone through the lecture of Dr. Alekha Das. It was wonderful, sir. We learnt a lot. And now again, we are going to learn about the career opportunities in pharmaceutical management. That is a very different topic and very useful for all the delegates today. Before we listen to Dr. Banerjee, it's my privilege to introduce Dr. Banerjee to the audience. Dr. Saurabh is a PhD in Pharmaceutical Sciences and M.Pharm. He is MBA with specialization in Healthcare Service Management. He has more than 18 years of experience in academics, research and industry cumulatively. Sir is currently deliberating academic responsibilities as Dean and Associate Professor in School of Pharmaceutical Management, IIHMR University. Sir is also sharing important responsibilities as Convener Board of Studies, SPM, Member in Academic Council, and as PhD Guide in IIHMR. Sir is also subject expert in the Board of Studies and PhD thesis evaluator in some leading universities of India. Felicitated with Pharma Leadership Award during the Pharma Leadership Summit organized by eHealth Eltes Technomedia Limited. Sir was conferred with Amity Excellence Award for Best Faculty in the area of pharmaceutical management at Amity University, Noida campus. Sir has received the Silver Jubilee Alumni Award for Prominent Alumni for the year 2020 from the Department of Pharmaceutical Sciences, Guru Jambeshwar University of Science and Technology, Hisar. Dr. Saurav has to its credit 22 research papers, 9 review papers, and 3 articles in international and national journals of repute. His current Google Scholar citation is 1635 with a H index of 8. A big round of applause to Dr. Saurav Banerjee for such an achievement. Sir is in the editorial review board of various national and international journals in pharmaceuticals, healthcare management, pharmacy practice, and pharmacoeconomics. Sir has guided more than 45 students toward dissertation of M.Pharm and MBA Pharm Tech and MBA Pharmaceutical Management Studies. Sir has delivered more than 30 talks in international MDPs and national conventions on various topics related to gene therapy, the next generation medicines, problem solution fit, product market fit, design thinking, access to medicine and diagnostics, free drug distribution policies, generic drug versus branded drug, sustainable brands, rational use of medicines, procurement and supply chain management of vaccines and essential medicines. Sir is a life member of Indian Science Congress Association and Indian Pharmaceutical Association. Sir's areas of interest are rational use of medicines, logistics, supply chain management of vaccines, and essential medicines. Quality assessment of drugs and pharmaceuticals and medical devices are other expertise. So we can say that Dr. Saurav Banerjee has a versatile platform, and I can say that whenever I talk to students about startups and entrepreneurship, I think that we have to do a little bit different out of the box thinking. And welcome to you, sir. We are eagerly waiting to listen to you. Please come on the dais and deliver your lecture. Please, sir.
Thank you very much, Prachi ma'am. Am I audible? Last bench, please. Am I properly audible? Thank you very much for such a illustrious, introduction. Glad to be a part of this esteemed, audience. And my sincere thanks to Falgun sir, Suhagia sir, Das sir, Mehul, and all respected faculty colleagues and my dear students. So, thank you very much, APP. My sincere gratitude to Professor Rajiv Daya and APP for giving me this particular platform. So, we have, learned professionals with us. Das sir talked about the discovery, but I am a totally different genre person. My job, so if you look at the life of a drug, right? So it starts from the drug discovery and development, right? Then you go for the toxicity studies and the clinical research. Third is the formulation development. And then the market is ready to put into the market. So I am that fourth guy. So I am going to, give some incidences and give you some latest, things which will be of immense value to you, and will try to, give you a perspective that what are the various facets of the drug when it goes into the market. What are the various areas of innovation, startups, and supply chain related things which are, associated with this particular drug. It's these areas are new, and my point of view of discussing this thing is that you can explore more about this thing. The area today is coming of the innovations and the startup. My presentation is going to cover some of the, new things, new ventures which we are, exploring into the area of pharmaceuticals. So here am I. Okay. So guys, how is the Josh? How is the Josh? आवाज मजा नहीं आया कुछ भी. How is the Josh? Wonderful, wonderful. Are you awake? Wonderful. बहुत अच्छी बात है. Okay guys, so give me 30 minutes of your life, right? and let me, give some, value to your your life, and after your 30 to 40 minutes, if I am successful, I will think fortunate that I have given something to the, audience. So I will try my level best to give something in this 30 to 35 minutes. First slide, please. So next. Guys, the area of pharmaceuticals is a very, very vast sector, right? We are in one of the, privileged sectors that you have other service industries like petrochemicals, textiles, telecommunication, but pharmaceuticals is a wonderful area, right? What are the various different areas in pharmaceuticals what we can explore? Number one area, as you know, are the area of the formulations, the tablet, capsule, syrups, injections, right? The entire gamut of formulations. The number two industry is a very, very important industry in pharmaceuticals, and that is the API industry. Okay, API is the core of the formulations, and entire, lot of career opportunities are there in the business development of API, regulations of the API, marketing of the API. So the second very important sector in the pharmaceuticals is the API sector. Number three sectors is a very, very emerging sector where the pharmaceutical industries need a lot of qualified people, especially with the background of biotechnology, biochemistry, microbiology, pharmacy, and is the area of the vaccines. When I talk of the vaccines, one part is the research and development of the vaccines. Second part is the development of the vaccines. The third part of vaccine is equally very, very important. That is managing the vaccines distribution in India. For example, when you saw the area of COVID, okay, the development of the application of the vaccine, COVID vaccine, it was itself a mind-blowing thing. And how do we, I have a short video for you that how are vaccines managed right from the government setup to the beneficiary, how does it reaches. So maintaining the cold chain temperature of the vaccines is itself a area of career opportunity for the pharmaceutical area. We'll explore it in detail, but vaccine is a very specialized thing. Number four area you might have seen in the COVID, nutraceuticals. What was the famous drug which came into existence in COVID? Nutraceuticals, Zincovit, right? Zincovit is by a very small pharmaceutical industry, that is Apex. Nobody has heard of Apex Pharmaceuticals, hai? Cipla, Lupin, Cadila, all these particular things. But nutraceuticals, export of nutraceuticals, Zydus has recently launched a department called the Zydus Wellness. The brand of Complan, hai? Complan is a brand taken by the Zydus Wellness, and lot of wellness products, vitamin C, and they are. So this is a very important area in the pharmaceuticals. You have the next thing which is going to rock India is the area of medical devices. You might have seen in little bit, a couple of months, Mansukh Mandavia ji, he has told that he wants India's, so current market of medical devices in India is around 15 to 17 billion US dollars, right? Government of India has given the target of touching 50 billion US dollars by, what is the time? 2030. Government of India is planning like anything for medical devices. This is the right time when the pharmacy students must come into the area of the medical devices. I will touch upon the medical devices, right from a digital thermometer to a surgical mask to a gloves to a high scan, CT scan, MRI, okay, and your cardiac stent, you put it in angiography, pacemaker, they are all the medical devices. They need people who have the pharmacy background, who can understand the medical devices into the area. I'll explore a little bit of this particular thing. Other areas adjoining with the areas are the diagnostic. You might have seen in the COVID, how was, Dr. Bilal Path Lab, Thyrocare, how they came up with these innovative startups, right? Testing of the COVID was a challenge for us. testing of COVID was a challenge. We do not have sufficient testing kits in India. We do not have PPP kits with us. We did not have, in vitro diagnostic kits. But today the scenario is changed, but still lot to do. Government of India has the vision that India should be self-sufficient in all these particular things. Merrill Life Sciences. Its startup is in, I think, in Gujarat only. Wonderful. They are making the cardiac walls, heart walls. You might have seen the tricuspid valve and bicuspid valve. One valve, artificial valve, is costing around, before Merrill came into this thing, they were made all by Abbott, made by Boston Consulting Group, all the MNCs. But today the scenario is changing. In one slide I told you what is the area of pharmaceuticals. Can you imagine this particular? I have not even, it will take me 1 hour to explain this particular thing. Let's move forward. Next. Next, please. Next. So I'm going to touch you little bit of things, when the product is launched into the market, right? Das sir explained the discovery part and the formulation part, but I am going to give you some aspects that what are the various important, avenues for you guys, pharmacy graduates, postgraduates, and where you can make your career into the various areas. And these are all the areas when the drug is launched into the market. Right. So the number one area, everybody, how many guys in this room are fascinated by the brands? Do you like brands? What? Give me some name of some famous brands. Yes, any guys? Any famous brands of phone or jeans or what? Yes, please. Yes. Anybody from the? Yeah. Give me the name of some brands. Allen Solly. Wonderful, sir. Or anybody else? Huh? Louis Philippe or Tesla. Wonderful. Tesla is coming. Brand is, I will not say the entire, population is driven by brands, but brand has a significant role in the consumer behavior, right? As the brands in the FMCG is there, it is very, 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 very important in the pharmaceutical field. Next, please. So brand management, the people, the good brands of pharmaceuticals, Digene, Gelusil, Volini, right? Dolo, right? You see, Dolo was nowhere. And suddenly, who were the brand leaders here? Crocin, Calpol, generation old brands. But suddenly, what has Dolo done? It's amazing. It's a case study to explore that how Dolo, perceived this particular brand. It is all about how do you put this particular brand in the mind of the physicians and the customers. Next, please. Some famous brands, So when we talk of the pharmaceuticals, what are the important areas? If you can easily bifurcate, one are the prescription drugs and the second are the OTC drugs. Okay. Prega News was one of the particular brand. There was a acute deficiency in the market. There was a complete gap. There was no product in the market for pregnancy testing kit. Mankind did a revolution here, and they find this is the power of market research. This is the power of research. Mankind did a very thorough research into the market, and they found the gap that there is no, they talked to the pregnant ladies and what are the apprehensions they are facing, and today Prega News is the #1 brand in the pregnancy kits. I have who made these brands? People like you who are sitting in this, audience, they are the brand managers. If you go on to the LinkedIn and search Gunjan Virmani, I know Gunjan, and she is the brand leader for Prega News. She is the person behind who has made Prega News the success in India, right? So similarly, you have very, very famous brands like Lactocalamine. Lactocalamine is by Piramal Foundation, okay? Very, very famous brands. So these are all the brands of the OTC brands. Next, please. Similarly, you have very famous prescription drugs. Next, please. India's largest selling top 5 prescription drugs. Lantus by Sanofi, insulin, okay. You have one almost top 5 brands, Augmentin, amoxicillin plus clavulanic acid. But Augmentin is number 1 brand in amoxicillin plus there are 5,000 brands who are competing in amoxicillin plus clavulanic acid. But how do you manage your brand number 1 in India? That is the career for you. Are you getting? Similarly, the area glycomet. Glycomet is simple metformin plus glimepiride. But how it is giving tough competition. I invited person from USV. He was a product manager for glycomet. They told that USV is such a fantastic organization. In your lifetime, if you get an opportunity to work as a brand manager in USV, must. He was working with Sanofi, but he was praising of USV. That is the hike what the companies have. Next, please. What are the roles of a brand manager? If so, you when you do pharmacy, you become R&D manager, you become production manager. Similarly, the person who makes the product a brand, he is the brand manager. What are the responsibilities of the brand manager? He has to be a very, very good market research. Market research, there are lot of organizations who do a lot of market research for the core pharma industries. Okay. What is market research? Because the core pharma industry that does not have the time to spend on this particular thing. For example, if I want to launch an insulin in India, it's a global product. The patent is expiring, right? I need a lot of information about the Indian market. Who is the prescriber? Who are the competitors? What is the disease profile? What is the epidemiology, right? From where you get these, information? They get it from the consulting organizations. In pharmaceutical companies like IQVIA, companies like WNS Global, companies like Deloitte, companies like McKinsey, they are the companies who do the market research. Nelson, okay, Nelson does a lot of research for the Garnier, the Lakme, okay. All these products, the market research is done by these organizations. They are world renowned organizations, okay. So these are some of the, like the brand manager has to be very good in the making the brand strategy. So these are the various profiles of the brand managers. Okay, next please. Required skills. If you want to become, want to go into this particular field, one of the important thing is that the starting point is that you must be able to read the reports. You can go to the site, to the IQVIA, go to the IQVIA. You will get a lot of reports which IQVIA has done about the market. Read these reports, okay. Your research acumen is very, very important if you want to go into the market research, and then you should have a very good creativity, okay. You might have seen wonderful advertisements on the, I remember some of the best Cadbury's ki ad dekha tha aapne, hai na? So creativity is again a very, very important thing what you need in these particular things. Next please. What is the hierarchy? If you want to become a brand manager, even if you are a fresher, pharmacy fresher, you do not have experience, you can join as a management trainee. Once you join as a trainee, you are, further you are extended to field executive, assistant product manager, brand manager, and I can, vouch on this particular thing, and from this group after 15 to 20 years if I come, I might see some of the vice presidents sitting out here. Dilip Sanghvi has become the managing director of Sun from a medical rep. Can you imagine it? I can give you 100 examples of this thing, the people who have risen from the bottom to the managing directors. That is the power of the pharmaceuticals. Next please. Madam Prachi is sitting here looking after the innovations. Guys, today the world is venturing into the e-pharmacy ventures. Lot of organizations are, converting from the traditional pharmaceutical systems to these e-ventures. Next please. If you want to get any information about any, for example, I want to take a antihypertensive drug. If I search telmisartan on the Google, okay, the first page which will be open, is of Tata 1mg. wonderful information, wonderful content. What do you need? They are giving you information about the disease, they are giving you information about the brand, generic version. What do you need? The first page is open of the Tata 1mg. Tata 1mg has, I don't know exactly, but employ numerable pharmacy. Who are writing this content for Tata 1mg? People like you. Who can write? Because you know pharmacy better, na? A person who does not has a pharmacy background, how he can write about the medicine? He cannot. Tata 1mg has rocked the space. And again I will come to the area of supply chain, that how Tata 1mg is, you might have seen that things have changed, Zomato, and so Tata 1mg is also doing the same. Okay, I will come to the area of supply chain when I explain this particular. But today, can you see this guy standing out here? Anybody who can tell me who is this guy? This young boy who is standing. I will not give you. His name is Arjun Deshpande. Yes, you might have seen when he introduced. He has opened a generic Aadhaar करके इसने एक business शुरू किया है. What he is doing, he is procuring the generic medicines from the manufacturer, and he has developed such a supply chain that he is delivering directly to the patients. Are you getting my point or not? The margin what you got, how does a pharma product move from the manufacturer to the CNF, from the CNF to the stockist to the retailer to the, you have particular margins, na? Everybody takes a margin. Why can't you, if you can get the product from the manufacturer to the patient, what Arjun has done? Please look at his profile of Arjun Deshpande. When he started this venture, he was giving +2 examination. He was 12th. Today he is the CEO of Generic Aadhaar. This organization name is Generic Aadhaar. This is the startup, What does the patient need? A patient, when he is suffering from COVID, what does he need? He needs access to medicine. Your research is of nothing if you cannot bring the research to the doorsteps of the patient. Marra insan to, what he will do? Try to do a small internship of 2 to 3 months or 4 months. Get into Tata 1mg. बोला सर ले लीजिए, तनख्वाह मत दीजिए मुझको, कोई दिक्कत नहीं है, मैं गुजारा दिस थायरोकेयर इन बॉम्बे. Now establishing a referral lab is around 25 to 30 crore rupees investment. You cannot set up a referral lab everywhere in India. This guy of Thyrocare, he had mechanism, जैसे हम कोरियर मंगाते हैं, he started this venture कि कैसे मैं blood samples को पूरे इंडिया से कलेक्ट कराऊं. What a remarkable. अच्छा, ब्लड जो है चिट्ठी विट्ठी तो है नहीं कि आपने जो है मंगा ली. Now blood is a very sensitive product. You have to maintain the temperature, you have to maintain the, otherwise your viability will be finished. He devised a particular mechanism through which he brought these blood samples to Bombay, get it tested, got all the reports to all the patients. What a wonderful thing. Similarly, SRL Diagnostic is pioneer in diagnostic industry. Okay. Similarly, Dr. Bilal. Dr. Bilal is the pioneer in Jaipur. He has his referral lab in Jaipur. Next, please. Friends, this is the future. Everybody knows Lupin Pharmaceuticals, hai na? But today, Lupin is venturing into Lupin Diagnostic. Lupin को क्या ज़रूरत थी? Lupin को कोई ज़रूरत नहीं है. Lupin is a global pharmaceutical industry. But Lupin has opened 4 to 5 referral labs in India. They are recruiting like anything. They need pharmacy people. They need people who have medical and life science background. क्योंकि उनको दूसरों को समझाने में उनकी जान निकल जाती है. Are you getting my point or not? Diagnostic is the area. Redcliffe Diagnostic, new venture. These are the startups who are, doing like remarkable. Pathkind. Anybody who has heard of Pathkind Diagnostics? Pathkind किसका venture है? Yes, absolutely. Mankind itself is a great example. Ramesh Juneja is, the Mankind is not very old and he just started 10 to 15 years, but today Mankind is among the top 5 in India. It is competing with the MNCs. It is competing with Abbott. Abbott is not a simple word. Abbott is throughout global. but mankind is competing. Pathkind is the venture of diagnostics. So diagnostic is also associated with pharmaceuticals. Are you getting my point or not? Next, please. Again, a lot of areas in the diagnostic industry, what you can venture. Next, please. I am going to spend some 5 minutes on the medical device because this is going to the life turning point of India. Government of India today, till today, आज भी जब COVID तो हो गया, this is a blessing in disguise. We did not have PPP kits in India. We did not have surgical masks. We did not have gloves. There are very few organizations who were manufacturing these things. 70 to 80% of the medical devices we were importing from outside India. We did not have any manufacturer in India who was manufacturing even cardiac stent. Cardiac stent आप लोगों ने देखा है? When you have a coronary blockade, you put a spring like thing, cardiac stent, pacemakers. Government of India ने आदेश दे दिया है, it started, but now it is on rock. Many of the states, गुजरात तो हर चीज में आ गई है ना? गुजरात, anytime if you happen to go make a industrial visit and go to Merrill Life Sciences, है ना? It is, I think it is near to Baroda only, right? Medical, next please, I'll just show you. Can you see this thing? This is the cardiac stent. This is the cardiac stent. You put it through a catheter in the blocked artery and then you inflate it and your cardiac, ये जो artery होती है ऐसे सिकुड़ जाती है ना? और इसको हमको dilate करना पड़ा. Otherwise तो अगर इसमें से अगर खून नहीं निकलेगा तो heart attack, खत्म कहानी है ना? Cardiac stent, pacemakers, right? So I'm just telling that by Government of India has given a mandate by 2030 we are going to touch 50 billion US industries. If you happen to, anybody from Andhra here, anybody who is from Andhra Pradesh, anytime if you happen to go and get a chance, go to Visakhapatnam and you have there the Andhra MedTech zone. गवर्नमेंट ऑफ इंडिया ने रॉ मटेरियल से लेकर फिनिश प्रोडक्ट से लेकर मार्केटिंग, सेल्स, एक्सपोर्ट, इंपोर्ट, रॉ मटेरियल सारी चीज एक ही जगह पे बना दी है. वो है आपका आंध्रा मेडटेक जोन विशाखापट्टनम. एंड दे हैव फाइनलाइज्ड मेनी ऑफ द स्टेट्स, हरियाणा, पंजाब, गुजरात, कर्नाटका. लॉट ऑफ मेडिकल डिवाइस मैन्युफैक्चरिंग कंपनीज आर कमिंग इन दिस एरिया. अभी अभी जस्ट अनाउंसमेंट हुआ है मध्य प्रदेश में उज्जैन के पास, है ना? They are going to make a very big medical device park. Guys, this is the time for you to venture into this thing. आप नहीं तो कौन? Who can understand the science behind the medical devices? You are the master. You know the science behind these things. You are the, you know each and everything about the drugs. इस आपसे बेहतर कौन मिलेगा? Venture into this, know about this area of medical devices much, because we do not have much of time, so I am not going in this detail. Next please. Some of the leading companies of the world की मानी हुई companies है Stryker. Implants you have seen, knee implant, hip implant, ये सारे जो है medical devices हैं. Stryker is one of the largest manufacturers of this world. Okay. You have Medtronics, number 1 and number 2 of the world. Medtronics, and you have the Boston Scientific. These are global companies. These are global companies. इसके साथ ही मैं आपको बता दूं कि इंडिया की बहुत सारी, for example, SMTs, Sajanand Medical Technologies, SMT, doing fantastic work. They are exporting medical, cardiac stents to outside India. Recently, I took the students to a organization which is Involutions. They are manufacturing startups, madam. Cardiac stent, they are manufacturing cardiac stents. आज से 10 साल पहले इंडिया में कोई था ही नहीं. If you happen to go to a hospital, कभी कैथ लैब देखिएगा जहां पर एंजियोग्राफी होती है. That is called as the cath lab. जो कैथ लैब का मशीन होता है ना, it is around 4 crore rupees. Involutions, this company, they are manufacturing these cath labs. This plant is in Bangalore. Okay. This is the area where you should venture. Next please. you are the people who know the regulatory and the clinical trials very well. Next please. Lot of opportunities are there in the clinical data management, because handling the data of clinical trials is itself a very difficult part, right? Selection of the patients, assigning them. So clinical data management is again a very important area. You can look into this particular area. Next please. Next please. Next. So next. So I was telling you about the consulting organizations, right? The market research, the Pfizer, Abbott, they need a lot of information. So they rely on the consulting organizations for a lot of providing these insights, because they have to take a lot of decisions regarding these things. So many of the pharmaceutical consulting organizations, leaders, WNS Global works for Glaxo, GSK, Roche, they work for Roche, GSK, right? IQVIA is almost number 1 in the world. Previously अगर आपने देखा होगा, it was IMS and Quintiles. IMS के पास दुनिया का सबसे बड़ा सेल्स का डाटा था, और क्विंटेल्स के पास सबसे बड़ा दुनिया का क्लिनिकल ट्रायल्स का डाटा था. तो दोनों ने सोचा, बोला अलग अलग तुम भी कमा रहे हो, मैं भी कमा रहा हूं, दोनों मिलके कस्टमर को लूटते हैं ना, क्या दिक्कत है ना? दोनों आपस में मिल गए और उन्होंने बना लिया आपकी IQVIA. फार्मास्यूटिकल कंपनी का कोई भी प्रोजेक्ट बिना IQVIA के डाटा के चल ही नहीं सकता. उनको IQVIA एक रिपोर्ट 1 करोड़ में बेचती है. The people who are recruited in IQVIA highly paid jobs, right? Area to venture, इसको venture करिए, इसको देखिए, right? You have similarly Clarivate Analytics, बहुत अच्छा काम करती है. These are pharma specific. Pharma is a Pune based company which does a wonderful work in, they work for US, Europe और India की भी. Novartis के लिए बहुत अच्छा काम करते हैं ना. ZS is one of the number 1 of sales and marketing consulting for, तो बड़े बड़े जो मार्केटिंग के प्रोजेक्ट्स होते हैं उसके अंदर ZS जो है आपका ये इंफॉर्मेशन प्रोवाइड करती है, राइट? स्मार्ट क्यूब, जेनपैक्ट, एक्सेंचर, आपको लगता होगा ये सॉफ्टवेयर कंपनीज है ना, बट एक्सेंचर हैज अ वेरी बिग लाइफ साइंस वेंचर. मेरे 2-3 स्टूडेंट्स एक्सेंचर में काम करते हैं, तो मैं बोला तुम क्या करते हो? बोला सर हमको टेक्निकल बैकग्राउंड देना पड़ता है, हमारा काम सॉफ्टवेयर का नहीं है, बट द इनपुट, व्हाट इज फार्माकोकाइनेटिक्स, व्हाट इज, इफ फॉर एग्जांपल दे आर मेकिंग अ एप्लीकेशन, अब उस एप्लीकेशन के अंदर अगर काइनेटिक्स यूज हो रही है, कौन बताएगा? किसी को आता ही नहीं. टी हाफ क्या होता है, रीनल क्लियरेंस टाइम क्या होता है, वही एमफार्म मेरे स्टूडेंट्स हैं, वो वहां पर बताते हैं. वहां पर बोला सर हमारा काम है टेक्निकल, हम ये नहीं करते कि हम सी प्रोग्रामिंग और आर प्रोग्रामिंग देख रहे हैं, वो सॉफ्टवेयर की टीम है, वो आईटी की टीम है, वो करती है. हमारा काम है टेक्निकल बैकग्राउंड देना. टेक्निकल तो उनको नहीं आता, जो आता है हमको उसी की पैसे कमा रहे हैं हम तो, कि नहीं? मतलब पैसे कमाने से है कि नहीं है? अच्छी तनख्वाह है. नेक्स्ट प्लीज. Next please. 1 or 2, sir, I have 5 to 7 minutes, right? Last I am coming to one of the very bright area, supply chain, है ना? तो हर कोई तो आज दीवाना है जोमेटो और स्विगी और, है ना, ऊबर का, है क्या है? What is this? How does Zomato works? How does Zomato works? How does Swiggy works? You order on an application, खाना कहीं पे बनता है कि नहीं? वो किचन है, वो मैन्युफैक्चरर है, और वहां से कहां पर आ रहा है? आपके हाथ में है ना. नेक्स्ट प्लीज. सो द ट्रेडिशनल सप्लाई चेन इंडिया फार्मास्यूटिकल्स के अंदर अगर हम देखें, वो क्या है आपका? मैन्युफैक्चरर से डिस्ट्रीब्यूटर या स्टॉकिस्ट, एंड देन स्टॉकिस्ट टू योर रिटेलर, एंड रिटेलर टू द. कैन यू ब्रिज दिस गैप? कैन यू मेक द प्रोडक्ट फ्रॉम द लाइक मैन्युफैक्चरर टू द, now there are big warehouses, right? Warehouse से सीधा आपका Amazon, I forgot the name of Amazon, है ना? Amazon has big warehouses, right? The moment you order the particular thing, the product is dispatched from that. अब वो तो रिटेलर के पास नहीं जा रहा है ना, आपके हाथ में आ रहा है ना. तो वो जो बोलता है ना फार्मेसी, वो डिस्काउंट जो दे रहा है, वो नाश्ता रहता है ना उसके अंदर, वो डिस्काउंट देता कहाँ से है? क्योंकि उसको मार्जिन बचा लिया उसने. वो मार्जिन बोलता है मैं पेशेंट्स को देता हूं ना 20% डिस्काउंट, मेरा पेशेंट भी पक्का होएगा. दिस इज द सप्लाई चेन. नेक्स्ट प्लीज. ओके, नेक्स्ट प्लीज. कैन यू जस्ट एक बार एस्केप करके वीडियो दिखा सकते हो? कैन यू कम टू द So friends, not only in the private pharmaceutical industries, सरकार गुजरात जो एंटायर जो मेडिसिन्स पूरे गुजरात स्टेट में आपकी डिस्ट्रीब्यूट होती हैं, हाउ डज द सप्लाई चेन ऑफ अ गवर्नमेंट हॉस्पिटल कम्स? वी हैव द बॉटम ऑफ आर इज द सब सेंटर. सब सेंटर के ऊपर होता है आपका प्राइमरी हेल्थ सेंटर, कम्युनिटी हेल्थ सेंटर्स, यू हैव द योर डिस्ट्रिक्ट हॉस्पिटल्स एंड देन योर मेडिकल कॉलेजेस. ये इतने सारी जगह पे मेडिसिन पहुंचती कैसे हैं? इट इज थ्रू द सप्लाई चेन एंड द मेडिकल कॉर्पोरेशन. गुजरात में गुजरात मेडिकल सर्विस कॉर्पोरेशन है, विच इज द एजेंसी विच हैंडल्स दिस पर्टिकुलर थिंग. सिमिलरली राजस्थान में हमारे है राजस्थान मेडिकल सर्विस कॉर्पोरेशन. आई एम गोइंग टू शो यू शॉर्ट वीडियो. आपको इससे पता चलेगा कि कैसे मेडिसिन जो है वो पेशेंट्स तक पहुंचती हैं. कैन यू ऑन द आवाज आएगी, इसके अंदर साउंड आएगा?
वर्तमान तथ्यों के अनुसार 40%
वॉल्यूम कर दीजिए थोड़ा सा.
रोगियों को उधार लेकर या संपत्ति बेचकर अपना उपचार कराना पड़ता है. WHO के अनुसार भारत की 65% आबादी अभी भी नियमित रूप से आवश्यक दवाओं से दूर है. 23% बीमार लोग तो कभी इलाज के लिए डॉक्टर के पास जाते ही नहीं, क्योंकि उनके पास इलाज कराने के लिए पैसे ही नहीं होते. इसीलिए मरीजों की पीड़ा समझते हुए राजस्थान की लोक कल्याणकारी सरकार ने मरीजों को निशुल्क दवा देने की अभिनव पहल की है.
2 अक्टूबर 2011 को राजस्थान के मुख्यमंत्री श्री अशोक गहलोत ने इस कल्याणकारी योजना का शुभारंभ किया. मुख्यमंत्री निशुल्क दवा योजना के अंतर्गत राज्य के सभी सरकारी अस्पतालों में आने वाले सभी मरीजों को सर्वाधिक उपयोग में आने वाली आवश्यक दवाइयां निशुल्क उपलब्ध कराई जा रही हैं. मुख्यमंत्री निशुल्क दवा योजना के अंतर्गत प्रदेश भर में कुल 15,000 दवा वितरण केंद्र स्थापित किए गए हैं. ये दवा वितरण केंद्र ओपीडी के समय खुले रहते हैं और मेडिकल कॉलेज या जिला अस्पताल में कम से कम एक दवा वितरण केंद्र 24 घंटे खुला रहता है. निशुल्क दवा योजना को लागू करने के लिए ड्रग परचेज, स्टोरेज एवं डिस्ट्रीब्यूशन के लिए राजस्थान मेडिकल सर्विसेज कॉर्पोरेशन का गठन किया गया है. योजना का सफल क्रियान्वयन व सुगम संचालन त्रिस्तरीय व्यवस्थाओं के अंतर्गत किया जा रहा है. प्रथम स्तर RMSC मुख्यालय, जिसका काम है प्रोक्योरमेंट. द्वितीय स्तर जिला स्तर पर औषधि भंडार गृह, जिसका काम है स्टोरेज. तीसरा स्तर अस्पताल स्तर पर औषधि वितरण केंद्र, जिसका काम है डिस्ट्रीब्यूशन.
राज्य स्तरीय RMSC मुख्यालय, इसमें कार्य संपादन के लिए बोर्ड ऑफ डायरेक्टर्स देते हैं नीतिगत निर्णय और टेक्निकल एडवाइजरी कमेटी करती है तकनीकी मार्गदर्शन. इसके अलावा कुछ विभिन्न सेल हैं, जैसे प्रोक्योरमेंट सेल, सप्लाई सेल, गुणवत्ता नियंत्रण शाखा, वित्तीय शाखा, लॉजिस्टिक सेल, IT सेल, कंप्लेंट सेल. कैसे पहुंचती है मरीजों तक दवा? सबसे पहले टेक्निकल एडवाइजरी कमेटी की सहायता से जो दवाएं खरीदी जानी हैं, उन आवश्यक दवाओं की सूची बनाई जाती है. इस समय 400 दवाइयां, 75 सर्जिकल एवं 72 सूचर्स क्रय कर लोगों को उपलब्ध कराने का लक्ष्य हमारे सामने है. WHO के अनुसार मात्र 350-400 आवश्यक दवाओं से 90% से अधिक मरीजों का उपचार हो सकता है. दवाओं की क्रय प्रक्रिया. दवा सूची बनाने के बाद क्रय प्रक्रिया का आरंभ राष्ट्रीय एवं राज्य स्तर के समाचार पत्रों में NIT का प्रकाशन किया जाता है.
क्रय आदेश. क्रय आदेश के प्राप्त होते ही संबंधित दवा कंपनियां अपना उत्पाद तैयार करती हैं और कंसाइनर्स के दिशा निर्देशानुसार प्रदेश के सभी 33 जिलों के औषधि भंडारों को सप्लाई की जाती है.
दवाइयों की प्राप्ति. दवा निर्माता आवश्यकता अनुसार दवाइयां व अन्य आपूर्तियां जिला औषधि भंडार भेजते हैं. प्राप्त दवाइयों में RMSC की मोहर एवं लोगो, नॉट फॉर सेल, एक्सपायरी डेट का अंकन, लेबल की सही स्थिति, फर्म की क्वालिटी जांच रिपोर्ट देखने के बाद डिलीवरी प्राप्त करता है। डिलीवरी प्राप्त करने के बाद क्वारंटाइन एरिया में रखी जाती है तथा ई औषधि सॉफ्टवेयर में इसकी एंट्री की जाती है। इसके बाद फार्मासिस्ट द्वारा क्वारंटाइन क्षेत्र में रखी औषधियों के प्रत्येक बैच से सैंपल इकट्ठे कर उसी दिन मुख्यालय पर गठित क्वालिटी कंट्रोल सेल भेजे जाते हैं। इस शाखा में सैंपलों से कंपनी का नाम इत्यादि हटाकर कोडिंग कर गुणवत्ता नियंत्रण हेतु एम्पैनल्ड रजिस्टर्ड लेबोरेटरी भेजा जाता है। प्रयोगशाला से सैंपल रिपोर्ट स्टैंडर्ड क्वालिटी प्राप्त होने पर सभी जिला औषधि भंडार को दवाओं को जारी करने के संबंध में निर्देशित किया जाता है। भंडारण यानी स्टोरेज। प्रयोगशाला से ओके रिपोर्ट आने के बाद औषधि भंडार का फार्मासिस्ट दवाओं को अल्फाबेटिकल यानी ए से जेड के क्रम में निश्चित रैक में रखता है। रैक में दवा इस तरह रखी जाती है कि उसका लेबल और एक्सपायरी डेट एक ही नजर में दिखती रहें। वो दवाइयां जिनको कम तापमान पर रखना होता है उसे ILR फ्रीजर अथवा कूल रूम में रखा जाता है। दवाइयों को अस्पतालों को भेजने की प्रक्रिया। संबंधित जिले के चिकित्सा संस्थानों से फार्मासिस्ट निश्चित समय अवधि में मांग पत्र एवं संस्थान की पासबुक लेकर जिला औषधि भंडार के कार्यालय पर आता है। मांग पत्र अनुसार फार्मासिस्ट दवाइयों को रैक्स में FIFO फर्स्ट इन फर्स्ट आउट सिस्टम से निकालता है और एक जगह एकत्रित करता है। फिर कंप्यूटर पर वाउचर तैयार करता है कि एक प्रति अस्पताल के सब स्टोर के फार्मासिस्ट को दे देता है।
अस्पताल सब स्टोर। अस्पताल के सब स्टोर में दवा पहुंचाने के बाद ई वाउचर के साथ दवा का नाम, मात्रा एवं स्ट्रेंथ का मिलान कर स्टॉक रजिस्टर में एंट्री की जाती है। इसके बाद दवाओं को रैक में ए से जेड के क्रम में व्यवस्थित रूप से जमाया जाता है। अस्पताल सब स्टोर द्वारा दवाइयां सभी संबंधित दवा वितरण केंद्रों, वार्ड, इंजेक्शन कक्ष एवं ऑपरेशन थिएटर को जारी की जाती हैं। जारी की गई सभी आपूर्तियों की एंट्री सब स्टोर के कंजप्शन रजिस्टर में की जाती है। दवा वाउचर की एक कॉपी दवा वितरण केंद्र के फार्मासिस्ट को दी जाती है।
दवा वितरण केंद्र। दवा वितरण केंद्र के फार्मासिस्ट आवश्यकता अनुसार दवाइयां अस्पताल के सब स्टोर से प्राप्त कर स्टॉक रजिस्टर में एंटर करते हैं। फिर दवाइयों को रैक्स में अल्फाबेटिकल यानी ए से जेड के क्रम में जमाते हैं। रोगी द्वारा दवा प्राप्त करने का तरीका। सर्वप्रथम मरीज अस्पताल के स्वागत कक्ष में पहुंचकर दवा कार्बन पर्ची प्राप्त करता है। इसके बाद वो पर्ची लेकर डॉक्टर को दिखाता है। डॉक्टर रोगी की जांच कर जेनेरिक दवाइयां कार्बन पर्ची पर लिखता है। फिर वो पर्ची लेकर अस्पताल में स्थित निशुल्क दवा केंद्र पर जाता है। वहां वो कार्बन पर्ची दवा वितरण केंद्र पर उपस्थित फार्मासिस्ट को देता है। फार्मासिस्ट दवाइयां निकालकर काउंटर पर रखता है तथा कार्बन पर्ची पर रोगी को देने वाली दवाइयों का उल्लेख करता है। रोगी द्वारा दवाइयां प्राप्त करने के बाद रोगी से कार्बन पर्ची पर हस्ताक्षर लिए जाते हैं। रोगी को ओरिजिनल पर्ची दी जाती है तथा फार्मासिस्ट डुप्लीकेट पर्ची रिकॉर्ड हेतु रख लेते हैं। दवा देने के बाद फार्मासिस्ट मरीजों के साथ दवाइयां लेने, रखरखाव के संबंध में काउंसलिंग करते हैं। वार्ड, इमरजेंसी व OT में काम में आने वाली दवाएं सीधे ही वहां के स्टाफ को इशू की जाती।
Right, so this was all about what I knew and something, and just wanted to share with you so that you explore these things. It's a bright opportunity for you in many of the various sectors. So like you have the R&D and the discovery, you have the clinical trials. Again, a lot of avenues are there where you can, when the drug reaches to the market, there are a lot of issues with the vaccines, with the drugs. You explore these particular area. I'm going to give this presentation, your faculty members. Please, sir, my request is that distribute to the students. Anytime you need any help, please connect me on LinkedIn, Saurabh Kumar Banerjee. If I know anything, Gujarat Medical Service Corporation is one of the finest in India. Gujarat or Rajasthan, they are very good in India. The pioneers were Tamil Nadu and Karnataka. Anytime if you need my help and if you want to visit the Gujarat Medical Service Corporation, I can help you in this particular thing. I am done with this thing. Thank you very much for your patient learning.
Thanks a lot, sir. It was really indeed a very useful presentation, very useful information to all the budding pharmacists who are just aspiring to becoming something different. And yeah, there are a lot of career opportunities that you have learned, like being a pharmacist, you can get into the various fields. Especially, sir, what resonates with me is the startups and entrepreneurship, because you know, when we work over startups, I'm very happy to share with you and sir, all the dignitaries that our university KPG is working great to work over the startups, and you know, it's a very general phenomena that I have observed in the recent years. Whenever I look after my incubator that gives support through Startup India Seed Fund, I received 1,800 applications all over India, and you will never believe 30% were pharma and wellness. Telemedicine, pharma, wellness, all those, and those really compete with the deep tech startups. And they take up the funds, they take up the mentoring support, and they're rising by leaps and bounds. So it's a great, you can see that it's a silver lining for all the pharmacists who can really convert their innovative ideas into startups and can become entrepreneurs. Thanks a lot, sir. Thanks, really, it's indeed a wonderful presentation. And just to let you know, our even 10 student startups have their own companies in Gujarat. So like it's a way like even students who are listening here or like sitting in the classroom few years back, now the CEOs, the directors of their own companies, because they had one idea that really changed the world, and they have given jobs to many. Actually, they are earning revenues, they are generating lots of employment to other people. They are giving jobs. A moment to you, please, I request.
Just because आपने startup की बात करी, many of you seen Shark Tanks India. आपने Shark Tanks में you have seen the guys who made a portable ECG device, Spandan. Have you seen that guy? That person came to our university and he is connected. If anytime you want, I can invite the person here also. We will be very happy to interact with the students. Yes, they have made a portable ECG device that is doing wonders. Spandan, आपने सुना होगा Shark Tanks?
Spandan, we have learned there, sir. It's very nice. Definitely, sir, we can call Spandan. And just to add those points, actually, you might not be knowing, our KPGU team has developed 2 startups of medical devices. So as you have like discussing, it is a endotracheal cuff pressure monitor cum controller for patients on ventilator. Another, you will be amazed, it's a real-time stress measurement response system. By measuring the interval between 2 N waves at milliseconds interval, we collect about 5-10,000 data of cardiac variability, and we predict the transition from sympathetic to parasympathetic nervous system, and just predicting how much stress a person has actually. And it's totally non-invasive device. So I just want to give a spark to you that these KPG team members are actually working over those medical devices. So yeah, okay, thank you very much, sir. Yeah.
चल रहा है, रख दिया नेम प्लेट. I request Professor Mehul Patel sir to take the seat on dais. नेम प्लेट रख दिया विवेक. Now I request Dr. Sailesh Koradia sir to welcome Mehul Patel sir with a bouquet. Our third session is on nano suspension, new development in long-acting injectables.
See at Sri B.M. Shah College of Pharmaceutical Education and Research, Modasa, Gujarat. and Master of Pharmacy from KLE's College of Pharmacy, Bangalore, Karnataka. He has secured first rank in his PG examination at Rajiv Gandhi University of Health Science, Karnataka. He has published more than 20 research papers in international and national journals and more than 40 presentations of his research work were presented at various international and national conferences as author and co-author. He is approved PhD guide at Dharamsi Desai University. Dr. Patel has guided one PhD scholar and more than 20 MPharm students for PG dissertation research work. His thrust area of research are micro-nano carrier-based drug delivery system, transdermal drug delivery system, nano drug delivery system, and herbal formulation development. He has hands-on expertise for quality by design, design of experiment, statistical analysis, and statistical optimization, etc. He is the life member of professional bodies such as APTI and APP. He is also a president of Gujarat State Pharmacy APP branch. With this brief introduction, I would like to invite Dr. Mehul to talk on nano suspension, new drug in long-acting injectables. Sir.
Thank you very much, Silas sir. Yes, I see that so many delegates are now actually searching for the venue of lunch, is it? Because my fight is with your hunger. Okay, I have to say what the topics that I have taken today as just 2 days back, I'm talking that this session I have to conduct due to some reasons, and actually I am here to fill this gap. So let me start with this session. First, before that, I would like to thank the APP, the yes sir, and the team of this KPGU that giving me this opportunity to talk on the this technical session on the nano suspensions, the field that we are working actually. So morning we have started with the Das sir sessions, wonderful sessions on this nano carrier system for the breast cancer. So this is what the area that is nowadays working. So many researchers and even the industries are looking for such kind of drug delivery systems or the technology. And ultimately, see, as Saurabh sir has already discussed about the brands. So these brands require some backgrounds. Okay, nothing is like that. No innovation is there, no new concept is there, no research is there. Then you will make the brand. It is very difficult nowadays. In previous day, it is okay that brands can be made by simply by what you can say that no innovation is there. But nowadays, some inputs has to be given. So research is one of the inputs that you have to give. Sir has already Saurabh sir told that market research. Same way, whatever the work you are doing, some kind of input you have to give. So these topics that given to me that is a nano suspension. Actually, my focus is on the this nano suspension, this technology or this carrier utilizations or use in long-acting injectable formulations. That is a new era in this long-acting injectables. In developed countries like USA and Europe, so many formulations nowadays with this LAI formulation, LAI-based formulations. These formulations, see, this long-acting injectable formulations, these are the meant for the name itself indicate long-acting. That means for the more number of days. Conventional formulations, they are generally for 1 day, okay, or half day or maximum 24 hours. Some of the transdermal like formulations are available for the up to week also, that is for the 7 days. Beyond that, that is for up to month or even more than the months, this kind of formulations can be developed. That means a single formulation, you can see this this graph of this LAI benefits. See, our oral formulations that is taken, that is given, and because of that, see, 1 day 24 hours, 24 hours, 24 hours, so many that rise or the decline in the concentration of drug in our plasma and bloods. So this sustained or extended release formulations, that is one of the good, you can say that, see, it is a when we are developing or doing any kind of research, is simple developing a sustained release formulation. It is not like that the average student should work on the nano carrier system. It is not possible. Okay, still there are some gap in the conventional, some problems and challenging associated with the conventional formulations. So SR is one of the alternatives that can be think of for the some of the molecules. So this LAI benefits the longer durations, that for the up to months. One of the formulations, that LAI based formulations, later on I'm showing the graph also, the data also, that is adherence. Adherence means adherence to the therapy. This is one of the challenge why this LAI is developed. So this adherence to the therapy is one of the requirements. Say example, I'll give the example of schizophrenia patients. So patient himself or herself is not knowing that whether I have taken the medicine or not. So what happens, they forgot that I have taken the medicines, or they deny that I will not take the medicines, they stop the therapy, sometimes they take the double dose also, because disease itself, it is having a such kind of tendency. So in that case, it is better that this LAI based the formulation is given. So once the formulation that is given by the trained personals or the skilled, that may be the physicians, and that will last, the one single formulation that will last for the 30 days. So no any other medications or treatments in between required for that duration of the therapy. So this is what the benefits of LAI. Say, whenever there is a benefit, there are some risk associated with this drug kind of drug delivery system. Some of the risks I have mentioned, not all. That is the termination of therapy, that is the removal is one of the challenge. Okay, it's because once it is given and some allergic symptoms has been occurred, so it is difficult to terminate the therapy. And one more problem, obviously all the sustained release or the extended release formulation having, that is a dose dumping. Those students are working in masters that might be knowing that the dose dumping is one of the challenge. So here the same thing is possible, because in these formulations in LAI, it delivers, we are delivering the 30 doses together. 30 dose combines in a single formulations. So what happen if all 30 doses are available in systemically? So obviously some we can say that some toxic reactions that may, some adverse reactions that may generated. So this is a risk associated with the long-acting injectable formulation. So it doesn't mean that we cannot develop. So we have to develop formulations that can overcome these kind of challenges. So this is what that the nanotechnology is one of the platforms that will provide, that will surety that this kind of risk may not be generated with the extended release or LAI based formulations. So see, conventional LAI formulations, because today I am going to highlight about the nano suspensions. So before that, what conventionally available? So it is not like that nano suspension, the nanotechnology come in the picture since last 20-30 years. Before that also, this depot based formulations are available. These are nothing but the LAIs, that is long acting injectable formulations. But this oily based formulation, when you see the compositions, I think, so these ingredients, even we have also worked with one anti-cancer agents, LAI formulations. In that, our rationale is that is to overcome these formulations. What problem with the formulations? The oil based formulations having some limitations. That is a pain at the injection site. Okay, because it is a depot means what? Depot is a very common terminology. Okay, I don't think it's required the explanation of what is a depot. Depot means in Gujarati we use word the depot. Depot is nothing but actually it is a depot. So depot means from where the distribution of this drug take place. Okay, the center, it is the one kind of center from where the distribution of drug take place. So this depot formation is occur at the site of injections, and that from that depot, the slowly, so how long? That is for up to 30 days, drugs slowly get released from the depots and that available systemically. So most of these depot formulations are intramuscular, deep intramuscular applications, and from that deep intramuscular application this release the drugs for up to the 30 days. So whatever that oil based formulation, see generally the oil aqueous media is always preferred when the injectable formulations we preferred. So oil based formulation sometimes that cause the pain at the site injection, some allergic, some sensitization reactions that may take place because the edible oils generally use coconut oils and sesame oils. So these oils, some of the patients are sensitive to this edible oils. So in that case also these sensitizations reactions that may occur. So looking at these situations, what is alternative? What is alternatives where we may not require the use of oils or some benzyl alcohol like solvents that that are irritating in nature? So alternative is the think of about some newer technologies. So what newer technologies? Newer technologies like say nanotechnologies is sir has already in the morning session, the first session discussed about the nanocrystals. So this nanocrystals or the nanosuspensions or this one more is the polymetallic barriers, that is a microsphere or nanospheres based technology that benefits or this technology can be utilized in development of LAI based formulations. So these advantage is what this nano suspensions and this nanosphere based technology, these are the aqueous based system. The previous system or the conventional system used that was the oil based system. So we are we are utilizing this technologies to get the same one better than in terms of the dose dumping and some sensitization reaction can be overcome. But the advantage is that even patient compliance is also given the priorities. So obviously this kind of, and why developed country has already adopted. I have graph also that what the actual scenario of LAI based formulation. So you can see that in 1960s, 70s and 80s in the green colors, that is a suspension solution in the sesame oil, that is are the oil depot formulations were used. Okay, later on in after 2000 it is found that microsphere based formulation has been developed, and currently the things that new formulations that is based on the LAI or that come in the markets, these most of the formulations are based on the nanocrystals. This blue colors, what different nanomedicines can be used in development of the LAI. You can see that I have covered all these nanomedicine in this graph. This is already published data what FDA approved in from 90 to 2015. So varieties of nanomedicines can be used in development. So each nano carrier having their own specificity, their characteristics and their advantage. They have some limitations like the neosomes or the liposome is there, the suspension, nano suspensions, microsphere, so many are there. And nowadays new words come in the pictures in the markets. Okay, every day some new words will come about the nanotechnology. So ultimately these all are the nanomedicines. Ultimately they are utilizing for the particular benefits. Okay, so we are focusing only this crystalline water insoluble suspension base. So why my today's session is I have not covered the actual technical. We have worked with two projects that is fulvestrant based nano suspension based formulation, that is also the LAI based formulation, and one more is that peprazole based brexpiprazole based formulation that is based on the microsphere. But more focus that you must understand the basics of technology, because see ultimately when you think about the some new words, when we heard that new words like euphosomes or liposomes, neosomes, first our reaction is what? This is not possible with our institute, or I can't do this. We don't have a technology. See, there's no big science behind this. Ultimately, your efforts, your intention should be there. You just go to the literature, you come to know that this is one of the simplest approaches, even that can be prepared in the laboratory. Yes, no doubt industries are using a sophisticated instrument, but that is at the manufacturing scale. Definitely some specific instrument is required, but something we have to just check, we have to evaluate whether this methodology will work or not. This methodology, this concept can be utilized for this particular development, for evaluation, or even some diagnostic purpose. So everything is possible. So we have to start with the situation. Yes, you have to work in directions, then only it will work. Otherwise, when we have started that projects on the LAI, the first question come in our mind: how we'll check the dissolutions of formulations? Should we check for 30 days? Because it is a 30 days formulations. When we prepare the simple tablets, what we think that we have to do the dissolution for 24 hours. Okay, 24 hours no dissolution, then also we think that I have to work for 24 hours for the dissolutions. So what about this LAI based formulations? Yes, definitely that is at the last stage. It has to be evaluated for a 30 days or whatever that's the requirement. But ultimately the basic requirement is the concepts. What concepts that you are utilizing for the developments of particular formulation, and that is very important. So this is a nano suspension. So what is a nano suspension? Nano different in terms of the conventional suspension that we will prepare in the dispensing pharmacy. We simply mix the drug particles, triturate in the mortar pestle, add the waters, suspending agents, our suspension is ready. Nano suspension is almost the same thing. Nothing difference is there. Same kind of ingredients, same kind of stabilizers, means suspending agents are used. The only difference is the particles or size of that solid particles should be in the nano size. So nano size is a requirement. Why this is a requirement? See what happens if large particles are administered intramuscularly. What problem? So many problems. Think from the formulation to after administration at the time of action also. See, if the first importance, the problem arise is syringeability. Whether we are able to penetrate this deep intramuscular, this large size particle? No. We require syringe and needles. Needle mathi this particle may pass, may sometime clog it. Okay, and even suppose example we have a large diameters needles, what happen after injections? It may cause a lots of irritations, pain at the site of injection, and that's why the first important patient compliance, considering the site should be the small, that is in nano range and the uniform. See, it is not the only this rationale behind the nano suspension. There are so many things, as sir has already discussed in the morning, that is the targeting can be achieved by the nano suspensions, then the dissolutions can be controlled by the nano suspension. So many things can be controlled, but this is one of the small things that we can consider while developing the nano suspension based formulations. So simply we have to this larger size particles that is converted in the smaller. Then see, this is what the advantages that we can get with the nano suspension. How to prepare it? Next questions. So we have two different technology. When you refer the articles of nano suspension, you'll find that whether it is a top down and bottom up and top down technologies. See, name itself indicates. See, we have technologies based on. See, generally three type of dispersion are there: moleculars, then colloidal, and the coarse dispersions. Okay, so nano suspension that come between the colloidal dispersions, that is in the nano size. So if we have a molecular dispersions, example is a solution. Drug is dissolved in a particular solvent. That means it is not visible. But even though we can say that it is a molecular dispersions, means drug is available at the molecular levels. So from that molecular level, molecular level is generally angstrom in size, 10⁻⁻10. So if slightly or some by technique or some by methods that we applied, from -10 to we have to bring it to -9 nano size. Okay, so that technology is known as a top down or bottom up. See, I have this one written here. Bottom up means molecular dispersion to nano suspensions. Okay, so we are increasing molecular size to, we are increasing to the nano size. So this is the technology that is I think we have prepared the crystals in the laboratory. Copper sulfate. You might have performed the practicals. So what is this? Yeah, no doubt. What the crystals that we are getting at that times that are the larger in size. But say example, we control by a particular methods to the nano size, then that will be the method for preparing the nano suspension. Very simple. Preparing crystal is the same method, that is the bottom up technique to prepare the nano suspension. And see, this is what is the main difference between these two? In laboratory, we always prefer the first method. Why? What the instrument required? What instrument we required? Nothing. We require only the beaker and solvent. And by heating or by cooling, whatever approach that we selected. Okay. But in industry people will not prefer because so many limitations. So they may think about another technologies, that is a top downs, means larger size particles. Sir has already in the morning session told that a big rock that is converted or reduced into smaller particles by means of some applied forces. Okay, so this is a technology that is used to prepare the nano suspension. I have listed, not going in details. You will refer the articles. You may find a number of methodology to prepare the technology. Some of the simple approaches I have shown here. One of the simple approach, that means crystal that we prepare by the solvent evaporation method or by the heating and cooling methods. So this is also one of the method that is utilized in preparing the nano suspension. This is, I think, emulsification method is there. Okay, first emulsion is prepared with the particular solvents, and by means of evaporations, you are getting a small size, nano size particles. See, the question arises in the evaluation section. I'm not sure whether I have covered it or not. That is the evaluation part. When this nano suspensions or the smaller size of particle is generated, this process parameter of this method that governs, that controls the size of particles. Okay, so that is a challenge that you have to do when you are working with the nano suspensions. Okay, I have seen that even in our institute also. I have seen that some of the guys or students, they have prepared 100 times, not 1, 2 or 10 times. They try for more than 100 times to get what? Nothing else, the size of particles. It is not like that you go to the laboratory and first instance you will get the nano suspension. It's a challenge because you have to govern everything. What governs? Simply by adding this another solvent in this beaker, or at what temperatures you are heating, at what rate you are heating, at what rate you are cooling down. All this kind of parameter has to be governed or you have to control. See, this is very important. Okay, because I have also observed that one of the students that working currently in same batch of MFAM, he is not even able to control the size. Okay, and I think we have only 2 months remaining for examination. Even in your case also the same thing. But the information you collect, say 100 times when you work with the nano suspension, and the informations that you gather by these temperatures, this size you are getting, this stabilizers, this problem, this method may work, may not work. By using this concentration, I am getting this size. So many informations and data you have. You are able to answer each and everything. Why not you have tried this? Why you have tried this? Everything you are able to correlate. So that is what the research that benefits to you. It is not like that in MPharm semester 4 or 3 and 4, the 6 months you are able to get some very big projects. It may also, but ultimately you have to get the knowledge, and knowledge that start initiated with taking such kind of research projects. So Bottom up, definitely the limitations, the most important limitation of bottom up technology is the method is generally not adopted by the large scale industries. Because in laboratory we have to prepare only for 5, 10, 20, 100 milligram, 200, 1 gram maximum. In industry, okay, that is in kgs, in even ton also. Depends on what size of the formulations. So that says, we cannot say that they use the big size boilers and the beakers for preparing this suspension. So they prefer the continuous method where they are getting, and that's why these technologies that media millings and high pressure, media milling is the best size process, but even a small size, small batch formulations, what we have prepared, that is full western based formulations that we have prepared by the media milling only. Even that is possible in the laboratory scale also. In the small scale also we can perform the media milling in the laboratory and we can generate the nano size particles. High pressure homogenization is one of the high pressure, high bar pressure instrument is used. That is the micronizations by applying high force pressures. Okay, it is simply it is reducing the size of particles. But when we do this media millings and this one more is that high pressure homogenization, the challenge associated with that is the energy of particles. As sir told in the morning sessions, the surface free energy, the energy carries by that particular charge. Okay, that because of that, these molecules are, you can say it is unstable molecules. Okay, these particles are unstable and they behave. Say example, today I have prepared nano suspension. That doesn't mean the same properties, same properties is after 7 days or after 1 month. Something changes may take place. Why take place? Okay, because they have energy. So whether they bind together, they repels, they attracts, something will happen, and that may lead to that some kind of innovations that we have to add. What innovations? Some small changes is required in developing. When you work with the nano suspension, you do some random trial and errors to find that yes, this particular work, this stabilizers, if I use a twin, it's not working. I work with the swan, yes, it is working. Why it is working? That you find out. Okay, I use elastidine, then this result I am getting. If I use this, then this result I am getting. So this is what the work we have to consider. So stabilizer is one of the requirements that we have to use, either stabilizers or the co-surfactant. As such, we are preparing nano suspension, you'll get it. You'll get the nano size. But how to stabilize? Because market requirement is what? Your product should be stable. Stable for 3 years, 4 years. For that, the stabilizer has to be used, or the co-surfactant. I think this is not the new words, because if the particle size is increased, say example, I have prepared the nano suspension today and I am getting the 200 nanometer size. Okay, then I pack it in the suitable containers. When at the time of administration, say example, this 200 nanometers, due to the aggregation, formulations will deliver for 30 days, then it may not be true with the size is changed. So size has to be remain same, or all the parameters and properties has to be remain same throughout the stability period. So that is possible simply by means of a suitable stabilizer has to be used. Okay, same way even the co-surfactants can also help to stabilize the formulations. So this is one of the summary of this methodologies, various methodology, which methodology can be used, when we can use this method, cannot be used. It depends on, it is not like that this solvent, anti-solvent may not work in the industry. It is not like that. Ultimately, important thing is whatever method you select, your product should be the stable. So what technologies? I think final few slides are there. What market scenario of this nano suspension based formulations. So there are few companies, they have already the patented technologies that I have listed already here. Nano crystal is that, Dizo cubes, nanopures, nanoAs. These all are the trademarks or their brand names that they have put in the markets. So they have developed, even you go to this website of these companies, they have put, they are providing the consulting also, that industries are hiring that particular technology in developing that particular formulations. I think I have put some images also. Yeah, see, these are the images. These are the nano crystal based products. Companies have developed, they have adopted the technologies that is developed by that particular companies. Even I have seen that nowadays companies are providing technologies for the liposomes and nanospheres, all nano carrier based consulting, or they are patented technology. What they have patented? What they have patented? Okay, they have patented the methodologies that they have adopted in preparing a particular carrier system. Okay, nothing new. A particular set of experiments they have run by particular compositions, and they are getting the good results in terms of the stability and efficacy, desired release and everything. So simply they have technology. They means they don't have API based technology. They have a methodology or carrier based technology. So if company want to prepare this peprazoles or anything, that particular molecules, okay, so that molecules that incorporated in that particular system, and that system is evaluated first, and then it is available in the markets. So these most of the nowadays what the formulations available. But say example, industries are spending the lot of money for adopting these technologies. Okay, what is the innovation? These are the innovations, nanocrystals and nanospheres, cubes that companies have developed the particular set of methodology. That is the innovations, and they are charging for that, charging for giving that particular technologies to the companies. So these are the nano base. See, this is some of the products nanocrystal base available in the markets. I have also put this Invesa, Trinza and Hafeza. This is one of the popular products in developed countries. Even in nowadays, these LAIs are available in India. Most of the formulations are available in the pre-filled syringe. Okay, most of these are the schizophrenia-based products, or sometimes nowadays even the anti-cancer molecules are also available for the long-acting injectable formulations. So these formulations are made for, see example, in our traditional or conventional system, the oral antipsychotics agents that is used, they are meant for the one-day therapy. That can be replaced by means of the 4 months, once a month for at least 4 months, and then 3 months injection cycles can be possible by means of these long-acting injectable-based formulations. So this is what the final conclusion of this today's session. This kind of technology is see, when you find out what is the cost of this therapy, just simply refer the websites. What is the cost? The cost may be the very high, but when you see about, it is simply replacing. Okay, when we compare with the price of this tablet with the, instead of that, one injection is given for the 30 days. Ultimately, it reduces the overall cost of the therapy, and there's a lots of the patient compliance. Okay, and one more important thing, this kind of dosage form has proven higher efficacy and improved therapy in terms of, because they are providing the continuous release for the longer periods. So when we develop a formulations for anti-cancer or the therapy where there's a no alternative, because in nowadays in the market when you find the price of the anti-cancer tablets, I don't think they're available in 10 rupees or 20 or 100 rupees strip. Even one tablets that may cost 300 rupees. I think it is a starting price. One tablet. So strip may be around 5,000, 6,000, 10,000. Okay, so for what they are charging? They are charging for the molecules. Okay, so preparing a molecule is not much costly. A simple synthesis. Okay, they are charging for their innovations, their inventions, their discovery. Okay, so this is what nowadays this LAI based formulation has been adopted, looking at even this is a prescribed also, even that is a preferred by the patients. That is reason that even though high cost, they have some good benefits in terms of the therapy, in terms of the stability, in terms of the patient compliance. So this is what the future of this LAI based formulations. So thank you very much. I think it is in time completion of the session. And if you have any questions that we can even discuss in later on session also, even poster session is there at that time also, we are available. So we can discuss out at that time also. Anything you want to discuss here, that also we can, anything else? I think some of have already left. the sessions. Okay, thank you. Thank you very much.
I know it's a lunch time, but till if there is any question from the audience side, sir is happy to answer your questions. Yes, please. Sir.
Yes, dose dumping can be.
Sir, please.
See, dose dumping is, yes, it is a challenge associated with all extended or sustained release formulations. So, when we prepare a formulations, we check what we evaluate. We evaluate that product behave. Say, example, when we perform the dissolutions, so we'll check the dissolution not one time. We'll take 1, 2, 3, 4, 5, 6 tablets. Why? See that whatever the methodology that we have adopted, it is a reliable. Okay, it is a validated that it behaves whatever you have designed. Okay, whether it is a 124 hours or it may be for the 30 days. Whatever the methodology that you have adopted, that's why simply I have seen that a simple example I can give is you prepare the tablets in the laboratory. Okay, what we do? Always we prefer that direct compressions or give the direct compression. Why? Because the methodology is simple. Powder, mix it, go for compressions. Okay. But have you seen that in that methodology, one of the most important requirement is a mixing. Is a mixing. If we not do the proper mixing, then dose dumping may take place. Because proper amount of polymers may not come in the pictures in that formulations, then tablet breakdown. And what we'll give the focus on weighing amount of drugs that it takes. Mixings, we are not doing mixing in the laboratory. No student will do. Will not waste time in that. We simply 5 times we rotate this spatula. Sir, mixing is done. We'll go for compression. Okay, because we have to finish the work. But see, these are the parameters that has to be evaluated. So ultimately that is in hand of the scientist. Okay, so in the hand of scientists that how we are preparing and how we are validating formulations so that such kind of risk may not be arise.
So thank you very much, sir, sharing your valuable knowledge with delegates. Thank you very much, sir.
I request Sailesh sir to give a memento to Mehul Patel sir. Thank you, sir. Dear delegates, we have now lunch break till 2:15 p.m. After that, the poster presentation session will start at 2:30 till 3:30. The presenting authors are requested to tie their posters according to their poster numbers, which are displayed outside the auditorium in a blackboard. Also in first floor and also in ground floor. After completion of poster presentation, all the delegates have to appear auditorium 1, in this auditorium 1 at 3:45 p.m. for the valedictory function. On behalf of all the committees, I would like to extend our gratitude to everyone present here and patiently listening all over the India for making this event more successful. Thank you.
saccharide and any other things.
So you have made by simple emulsification.
Emulsification technology. For that purpose, hydrophobicity improve as well as hydrophobicity improve, emulsification property improve. Then the thermoreversible material you can use as a.
What are the advantage of that thermo-reversibility in case of your nanotechnology?
Nanotechnology. Actually, my purpose is microsphere, and this microsphere we can prepare by temperature factor, with the help of a temperature factor. Whenever we form a solution at 80 degree, then it melted, completely solubilized, then down the temperature and putting the beads in the watery solution. Then these beads at the room temperature, it become a microspheres. That's why I use the thermoreversible gallon.
Sir.
Sir, today I am here to present about my research work on formulation, development and categorization of ninted and bloated micellar drug delivery system for improved therapy of idiopathic pulmonary fibrosis. So my target disease is the idiopathic pulmonary fibrosis, which is a type of lung cancer. So it is the chronic lung disease. Then scarring occurs in lungs. Then it is a cause is unknown. Then more common in men than women and more common in older.
What is the rationale of your studies?
Okay, sir, rationally is जो बायो अवेलेबिलिटी है जो निंटेड आणि ड्रग का वो 4.7% ही है सिर्फ, तो उसके लिए हम उसको नैनो कैरियर में डाल के उसका जितना हो सके उतना बायो अवेलेबिलिटी इंक्रीज़ करने का ट्राई करेंगे.
चेक द बायोबिलिटी.
सर उसका जो मार्केटेड फॉर्मुलेशन है निंडानिप करके ग्लेनमार्क फार्मा का है, उसमें वो लोगों ने उनके पेटेंट में लिखा हुआ है 4.7% इसका बायो अवेलेबिलिटी है.
You have done this project to improve the viability.
Yes, sir.
Isn't it? Have you checked that your project have useful in increasing the viability of ninted?
सर हम लोगों ने उसका फर्दर जो डिसोल्यूशन प्रोसेस किया है इन विट्रो एंड उसका सेल लाइन स्टडी भी किया है, तो उससे हम लोगों को पता चलता है कि ऑब्वियसली उसका इंक्रीज़ हुआ ही है, क्योंकि हम लोगों ने सेल लाइन स्टडी किया. नॉर्मल जो सेल है ये नॉर्मल सेल है सर, और देन ट्रीटेड विथ ओनली ड्रग डिस्पर्जन इन डिस्टिल्ड वाटर.
These are not the study to which can prove that the viability have been increased. Isn't it? So when you are targeting something, na, you have to prove that target.
सर हमको सिर्फ बायो अवेलेबिलिटी इंक्रीज, तो मतलब हम लोग है ना इम्प्रूव थेरापी कर रहे हैं. जो मार्केटेड फॉर्मुलेशन है उससे इम्प्रूव थेरापी हम लोग देना है, और वो अभी जो मार्केटेड फॉर्मुलेशन
पास से प्रोडक्ट लेकर जाएंगे.
यस सर, श्योर. हम लोग जब उसका पूरा करेंगे, एक्चुअल में सर ऐसा है कि जो ग्लेनमार्क का है वो ओरल टैबलेट है, और हम लोगों ने इसको ड्राई पाउडर इनहेलर करके फ्रीज ड्राइंग इंस्ट्रूमेंट से हम लोगों ने डायरेक्ट उसको DPI थ्रू देने का सोचा है. आगे सर प्रोसेस उसका चालू है, अभी वो फाइनल नहीं किया, बट हम लोग DPI थ्रू ही आगे जाने वाले, तो उसका डायरेक्ट जो टारगेटेड डिलीवरी होगा.
सॉल्युबिलिटी ऑफ दिस ड्रग?
सर सॉल्युबिलिटी इसका वाटर में, सर वाटर में 0.0005 समथिंग है.
What is the reason कि ये इसका viability कम है?
सर उसका रीज़न ये है कि ये इसकी लो सॉल्युबिलिटी है एंड उसकी.
You can improve this. आप क्या कर रहे हो, डायरेक्ट viability पकड़ रहे हो.
हाँ जी सर.
You are not finding कि viability क्यों कम आ रही है.
हाँ.
You just find out the reason why there is.
सर उसका सॉल्युबिलिटी कम है.
लेकिन आप मेथड तो solubility improvement की कर रहे हो ना, तो solubility तो स्टडी करो पहले. इसका जो फेस solubility स्टडी होता है वो परफॉर्म करो, उसके बाद में.
ओके सर, श्योर सर, वी ट्राई.
उसके बाद में.
ओके सर, श्योर सर, वी ट्राई ऑन दिस.
151. Research your
research sir.
Monoclonal antibodies and nucleic acid, etc. So these are the devices are available for. Devices are available for delivery to the biologics. So these are some limitations of biologics.
So what are the limitation of this intestinal
Majority
patch system? What are the disadvantages of this intestinal patch system?
Intestinal, वो कभी कभी क्या होता है जब इंटेस्टिनल हम पेस्ट सिस्टम को डालेंगे तब वो स्टिक भी नहीं हो सकता, या खाना खाने के बाद डालेंगे या पहले डालेंगे उसका इश्यू आ सकता है सर। और ये
किसका है? चला હવે થોડા જનરલ મારી લો ને, તમારી પાસે રાખો, જેને આપો ને સારું હોય ને.
So myself Siddharth Thakkar, and the topic for the poster is the human on a chip. So basically it is the concept of the organ on a chip. So till now we are using the for the drug testing we are using the animal models and the other in vitro cell cultures, but basically it is having several disadvantages that animal models cannot perfectly mimic our human condition, because it is our human complexity is very much. And even if we are using the 2D cell culture organoids kind of external cell culture, so that is also not able to predict the drug safety and efficacy. So for that, this is the concept of organ on chip came. So basically in the organ on a chip we are using the concept of microfluidics, in which we are manipulating the fluids at the micron levels. And basically suppose in the organ on chip, if we specifically talk about the lung on a chip, so it can mimic the
preparation method. So for sample preparation method, there are various extraction processes, whether it is solid or liquid, and then
तो एम कवर कर ले बच्चा પછી લાઈવ મૂકી દે.
ना चंद्राची चंद्राची ग नवराई माझी नवसाची नवसाची ग अप्सरा जशी इंद्राची इंद्राची ग अरे देवुड़ा లోకటే పిల్లడాంట దగ్గరై నన్ను చేరదీస్తివే బుట్టబొమ్మ బుట్టబొమ్మ నన్ను చుట్టుకుంటివే జిందగీకే అడ్డబొమ్మై జంట కట్టుకుంటివే బుట్టబొమ్మ బుట్టబొమ్మ నన్ను చుట్టుకుంటివే జిందగీకే అడ్డబొమ్మై జంట కట్టుకుంటివే
Thank you all. Super performance. Now for the next performance, I like to call Shikha Jadia Sam for B Farm KSP KPGU.
Hello. Good afternoon, everyone. Myself, Shikha Jadia from B Farm second year, semester 4. Today I'm going to present one bandish which belongs to Raga Vrindavani Sarang, which is very ancient raga from Indian classical music. Thank you. Motu Motu. Bana bana dhoondana bana bana dhoondana Sita ko छिपा गाए कृष्ण मुरारी बाना बाना ढूंढना जाओ शीश मुकुट और कानन कुंडल शीश मुकुट और कानन कुंडल बंसीधर मन रंग फिरता गिरधारी बन बन ढूंढना जाऊं बन बन ढूंढना बन बन ढूंढना बन बन ढूंढना जाऊं Thank you.
It's amazing performance. Thank you, Shikha. Now I request all the dignitaries to take their seats on the dais. Honorable Provost of KPGU, Dr. A.P. Chaudhary sir, speakers Professor Alekha Das sir, respected Director KSP, Dr. Falgun Mehta sir, Shailesh Koradia sir, Professor Dr. Jigar sir, Mihir sir and Prachi madam. Prachi Madam, please come. Now I request our Director Sir KSP Dr. Falgun Mehta Sir to welcome Honorable Provost Sir Dr. AP Chaudhary Sir with flowers. Okay, thank you. As we draw near the conclusion of this remarkable conference in pharmacy, it is with a blend of gratitude and inspiration that I stand before you today. These few hours have been a testament to the power of collaboration, innovation, and dedication within our field. First and foremost, I extend my deepest appreciation to all the speakers, jury members, presenters, and attendees who have contributed their expertise, insights, and perspectives to this gathering. Your contributions have enriched our discussions, broadened our horizons, and propelled us forward in our collective pursuit to excellence in pharmacy. Without further delay, let us begin this momentous occasion with gratitude and sense of accomplishment. I request Dr. Jigar Vyas Sir, Professor KSP KPGU, to give concluding remarks. Jigar Sir.
Thank you, Madam. Honorable Provost Sir, Director Sir, Speaker, other dignitaries, my colleagues, and dear participants. So as we know, now we are on the verge of completion of this event. So let us see some summary of these events. Well, talking about the history, the seed of this event was sown back few months back, and registration was open in January. And we have got a truly overwhelming response to the extent that we need to prepone the date of the submissions that is due to your passion and your overwhelming response, dear friends. We have received the participation from across the India, including many states, and even beyond the India, we have received some participation from globe, maybe Australia also. You may not believe, we have started with the expectation of 150 offline posters and around 100 online posters. But your passion and dedication has surpassed all the expectations, and we have received around 169 offline posters only in the subject of pharmaceutics, and including others, total 250 offline posters that you people have presented today and we have evaluated. And in the online also, we have received equally impressive numbers, around 115 online posters, which we have completed already yesterday with the 5 different jury members. So let us not forget the invaluable contribution of our jury members. For this complete event for evaluation, we have invited 18 jury members from 14 different organizations, and 5 of them have joined online and evaluated online posters. 13 of them have joined today to evaluate our offline posters. Their expertise for jury members and dedication has ensured very rigorous but a fair selection of today's posters. Dear friends, as we are on the close of the day today, let us take with us the inspiration gleaned from these enriching interactions. Let the knowledge exchange today fuel our collective pursuit of excellence in our chosen fields. Remember, dear friends, the collections you have made today are just the beginning. The discussion that took place here should resonate in our future endeavor, and may the bonds formed here lead the lasting collaborations. Thank you once again everyone who contributed in making this conference not just successful but a historic. We look forward to reuniting in event in future also with more stimulating discussions. Until we meet again, let us continue to strive for excellence and innovations. Thank you very much, friend.
Thank you, sir. Now, the time for announcement of the winners. So for that, we arrange this competition online and offline both. So first, I will start with the online winners. In that first, pharmaceutics department, runner-up is Rujuta Giri, JSPMS College, Pune. Second one is Sumitra Devi Chaudhary, Kale College of Pharmacy, Hubli, Karnataka. Second, chemistry and QA department, Khushbu Patil, MIT University, and Yogesh Mahadev Beats Pilani from Cognosy and Ecology department, Savarna Roy, Birla Institute of Pharmacy, and second from the same college, Tanishq Saini. Have a great round of applause for all the winners and very congratulations to all. Now it's time to declare the offline winners. For that first, Sudics department, I declare their names. Participants are requested to come on the stage and collect their certificates. May I kindly request Honorable Provost Sir KPGU Dr. E.B. Chaudhary Sir to present the certificates. First, for the pharmaceutics department, we have two winners at the third rank. So start with the third rank. First, third rank, there are two winners. In that, one is Aishwarya Nair, Parul University. Third position again, Ashwini Patel Madam, KSP KPGU. Sir, Ashwini Patel Madam. For the second position, Manika Menon, ITM. Be ready, Manika. For the second position, Manika Menon, Madam, ITM. And the first position, Krupal Sadhu, KSP KPGU. Thank you, sir. Now I invite Professor Alekha Das sir to present the certificates. From the Ecology and Cognosy department, runner-up is Patel Meet ITM. For the first position, Praveen Kumar Paswan, KSP KPGU. Thank you, sir. Now I request Director of KSP, Dr. Falgun Mehta, sir, to present the certificate from QA and Chemistry Department. Runner-up is Dr. Upasna Yadav, Madam, KSP KPGU. And the winner is Dr. Pinkal Patel, Parul University. Thank you, sir. Have a great round of applause for all the winners. Congratulations to all the winners. As we depart from this conference, let us carry forward the momentum and spirit of collaboration that has defined our time together. Let us continue to exchange ideas, foster partnerships, and push the boundaries of what's possible in pharmacy. Let us remain steadfast in our commitment to evidence-based practice, patient-centered care, and the highest standards of professionalism. Now it's time for a vote of thanks to cheer the efforts of all the persons to make this event successful. For that, I would like to call Dr. Mihir Parmasar, Professor KSP KPGU.
Thank you, ma'am. Good evening, everyone present over here and online. I, Dr. Mihir Parmar, feel very privileged to be able to present here on this occasion and asked to deliver a vote of thanks on behalf of Krishna School of Pharmacy and Research, Dr. Kiran and Pallavi Patel Global University, at today's Indo-US International Conference. As we know that saying thank you is a respectful way to show appreciations towards a persons, but these words would be less for our today's chief guest, Professor B.N. Suhagia sir, our speaker, Professor Alekha Das sir, another speaker, Dr. Saurabh Banerjee sir, and Dr. Mehul Patel sir. They are specially here to deliver a talk onto the emerging topic in the research. Their talk not only give us a knowledge about a recent area into the research, but they taught us how to think like researcher or a scientist. Thank you very much, sir. Next, I would like to thank you, our director, sir, Dr. Falgun Mehta, sir, because of whom this conference was possible. He played the role of the backbone to us and helped in the organization of this event. A special thanks to all my teaching and non-teaching faculty for a grand success of this conference. Today, almost more than 300 participants has presented that poster offline mode as well as into the online mode yesterday. I want to extend my special thanks to the management, to Honorable President Sir, Dr. Jagdish D. Patel Sir, who has been always been supportive in every task which is asked by the Krishna School of Pharmacy. I want to extend my special thanks to the Provost Sir, Professor A.B. Chaudhary Sir, Registrar Sir, Dr. Chirag Nagda Sir, OSD Ma'am, Palak Ma'am, and Deputy Registrar Academy Sir, for their time-to-time help and encouragement. I also want to thanks jury member, photographer, storekeeper, transport department, computer networking, food department, volunteers, peons for their extraordinary work to us to make this conference a grand success. Hope everyone enjoyed it. Thank you once again, all of you. It was because of everyone hard work this event was possible as a huge success. Thank you very much. Vande Matram. Jai Hind.
Thank you so much, sir. All participants are requested to fill the feedback form link, and the link is shared in the group. Together, let us embark on the journey ahead with renewed enthusiasm, unwavering determination, and a shared sense of purpose. The future of pharmacy is bright, and it is ours to shape. Thank you once again for your participation, your patience, and unwavering commitment to advancing our noble professions. Let's over this session with the national anthem. All are requested to rise for the national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उज्जल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
जय हिंद भारत माता की जय. Participants are requested to collect their certificates from the desks at outside, and all are invited for the high tea at the dome area. Don't forget to fill the feedback form. The link is shared in the group. Thank you.